Hello everyone. Dr. Sujit, we can start live, so we can start the session. Sorry, we are not audible. Hello. Yes.
Audible?
Yes, now it's audible. Yes.
I welcome Dr. Vipin Verma for this national conference. Dr. Vipin Verma is Program Director with BioGlove Research and Solution, New Delhi, based biotechnology organization founded by him. He holds a PhD degree in tissue engineering from IIT Delhi, from where Dr. Bismita also did his degree. And PGP in International Business from Indian Institute of Foreign Trade, New Delhi. Before BioGlove, he was application manager in Corning Life Science and application scientist with Becton Dickinson BD Bioscience, India, and research officer in AIMS, New Delhi. He received industrial training in bioprocess and industrial cell culture from USA, Korea, China, and Japan. Dr. Verma is experienced in 2D and 3D animal cell culture, organ culture, stem cell biology, flow cytometry, ADME tox assay for drug discovery and development, molecular biology, medical microbiology, biomaterial, and tissue engineering. He has been a lecturer of biotechnology at Amity University, Noida, and a guest faculty of tissue engineering at Manipal Institute of Regenerative Medicine, Bangalore, India. So I invite Dr. Verma for the talk, Artificial Intelligence in Drug Designing and Discovery. Please, Dr. Verma.
Thank you, Dr. Sujit, for the introduction. And I would also like to thank Dr. Bismita for inviting me on this platform and with the theme, which is really very relevant today. In the morning, we saw the beautiful presentation by Dr. Veena, and she captured beautifully about the way drugs are made and they are manufactured, starting from the conception till they come to your mouth. So it has made my life easy for this presentation, and I'm touching upon a topic which is the artificial intelligence. Now, this is a growing area which is coming up, and we all are seeing the
Dr.
application.
Verma, it is
Verma,
not audible properly.
So
Sorry,
that's what I wanted to also mention.
Not audible. We are not audible to you.
not audible.
Slow, very slow.
A little bit sound is needed at your end. Either you enhance your sound or you just keep the
Volume
mic.
thoda बढ़ाइए.
Okay, you give me 5 minutes, and I'm getting my headphones so that you can have a better audio. Yeah.
Okay, thank you.
Okay, just a minute. Hello.
Yes.
Am I audible now?
Yes. Clear.
Okay,
Yes.
thanks.
Yes.
Yeah, thank you. So should I start with the, beginning
Yes.
or?
From where you left, you can just start.
All right. So I was telling that in the morning what Dr. Veena has presented. She has given a very good picture about the drug discovery program, and I'm sure the people who are attending this lectures and who are not aware of much about the process of discovery, they have got a very good idea. right? So what I'm touching upon today is the artificial intelligence in drug discovery. Artificial intelligence, it's a growing area, and I mean, we have experiencing it on day-to-day basis, which I will, tell you in my presentation. And this area is a bit complex also, so we need to have a good understanding about the languages, about the computers, the bioinformatic systems, and the softwares and all those things. So what I've done in my presentation is that I have used the minimal jargons, and I have tried to make this presentation as simple as possible, so that people who are attending this seminar and who are not, our young minds who are attending here, they may at least carry some portion, and they can, get some idea about it. So I'm presenting uh, my presentation here. Just a minute, let me share the screen. I think, Dr. Bismita, can you please enable me to share my screen?
Just morning also that was a problem. So I think if I share screen from my end, it will take my screen. Uh,
You have to give me permission.
in the morning, it we assembled it.
ଏଇଟା ଦେଖ ତିନିଟା ଡଟ୍ସ ଅଛି ସେଠି ସେଠି
Can you try now if it is coming?
Yes, it is coming now. Okay, so can you see my screen now?
Yes, yes, it's audible, it's visible, everything. Please proceed.
Okay, thank you. Okay, so what I'm discussing today in my presentation is what is artificial intelligence? How does it work? How artificial intelligence is used in drug designing and admitox studies. And then I will be touching upon few ethical aspects of artificial intelligence. So I start with some, questions here because it's not interactive. That's okay, but I'm sure people must have used the smart assistants. They must have used like Siri or Alexa. So if I am saying, "Hey Siri, what is the date today?" So immediately Siri will start answering. Or if I ask Alexa, "Hey Alexa, what is the weather?" You know, so they will start telling us about what is the current weather, and they will also give you the wind directions, and they will also give you, some additional information about that. So you must have used the spam filters on email. When we use the Gmail, you have some spam filters, and Gmail automatically it filters out certain promotional emails or some non-essential emails which are coming as a marketing tools from different organizations, and it puts in a folder. We call it as a spam folder. And when you look at the spam folder, you will find that most of the emails, they are the totally useless emails. So it has got some sort of, training that, it can filter out the emails from all your inbox and then keep it in a separate box. So that is one. Other is song or TV show recommendations from Spotify and Netflix. I'm sure people are using here, almost all people are using the Netflix or, the streaming platforms, which we call them as a OTT. And whatever you are watching, Netflix recommends, based on your history. So, or whatever you are listening, like Ghana.com or something like that, then they, these portals, they start suggesting you that what you should hear next, or what should you should watch next. So that is also, one of the examples. And then there are conversational bots for marketing customer service. When you are visiting a particular website for buying some things, or for some other maybe you want to buy some Amazon products, or you want to go for Flipkart, and if you want to get a help, then definitely you may see that there is a pop-up which is a customer support chat box. And when you type something hello, so within a fraction of second they will say hello. So it is not that that somebody is just waiting for your hello and he or she is typing hello, but it is a kind of conversational bots where they have pre-fed instructions, and based on your hello it will recognize hello, and then it will start conversation with you. So, and then Facebook, YouTube suggesting you videos, you are watching Facebook, you are watching YouTube, and then so immediately you start seeing some suggestions that you should watch this, you should watch that, and you realize that it comes of your interest. Whatever they are suggesting, most of the time it is of your interest. Now, why it happens? Because it is taking the cues from your history, what your watch history, and then they're suggesting you. So if you have used at least one of them, you have experienced artificial intelligence. These are all the examples of the artificial intelligence. Now, when we say artificial intelligence, it is the intelligence which is demonstrated by machines. And using artificial intelligence, we make machines intelligent enough to mimic cognitive functions that humans associate with the human mind. For example, learning and problem solving. So we are putting all these properties, all these characteristics, and we're trying to put them into a machine. So that is the artificial intelligence. Now, if you see here, this is our artificial intelligence. It is helpful in decision making because you provide certain data. It can solve complex problems. It can perform high level of computations, which a human brain can do, or maybe beyond that. And it has also increased the accuracy of the result. Day by day, it is increasing the accuracy of the result here. So I would say that more and more intelligence is coming up. So, as per the Nick Bostrom is a philosopher at the University of Oxford, and there are three major levels of development of artificial intelligence. And he wrote a book which is known as Super Intelligence. It's a good book, and he has defined the three different types of, or the three stages of the artificial intelligence. The first is the artificial narrow intelligence. Second is the artificial general intelligence, and third is the artificial super intelligence. So artificial narrow intelligence it has already incredible pattern recognizing abilities. So we are using it for I would say for decades. So in huge data sets, which makes it perfect for solving text or voice or image based classifications and some clustering problems. I'll give you an example here. For example, people, you must have heard that the supercomputer has won the chess championship against a human, or a particular like Gary Kasparov or Anand Vishwanathan, they have played chess with the supercomputer. So this is a kind of the artificial narrow intelligence. So it is an algorithm that can excel at a precisely defined and a single task. And this is there in our in our use since decades, 20, 30 years back. The people have started working, or I would say from the time of the computer research. So this artificial narrow intelligence is always there. Then comes the artificial general intelligence. Now this general intelligence is a human level artificial intelligence. And with this artificial intelligence, we can reason, we can argue, we can solve issues, right? And this is a really, the way we believe or we behave in the same way the machines are expected to behave, or they have to, work like that. It is a possibility in the near future. And when we say near future, as per the author of the book, it may be within 50 years, or maybe earlier. Then comes the artificial super intelligence. It is theoretically what could have humanity's combined cognitive capacity or more. It's obviously, would not be able to grasp its knowledge and understand its reasoning. And most important part, it is not a desired stage to reach, right? Nobody would like to reach at this stage. Otherwise, what will happen? That the machines are going to dominate the humans. You must have seen the movie like Terminators. You must have seen the Rajnikant movie like Robot, where the it has been shown that how the machines can empower or overpower the humanity. So that comes under the artificial super intelligence, and that's the reason it is not a desired stage to reach. Now, if we see this picture, so this is the artificial narrow intelligence, and this is its middle point. This is the artificial general intelligence, and here it is the middle point. And this is the artificial super intelligence, where here it is the middle point. Now, this green dot, it is showing what is the ideal artificial intelligence should be, at what place it should be. So you can see here, we don't want this green dot to reach here, because then the machines will be equivalent to human beings, and maybe there will be a man machine conflict. And definitely we don't want this green point to come or cross this barrier also. Otherwise, what will happen here? The machines are going to basically power us. They are going to you dominate us. So this is the ideal place. So green dot indicates a theoretical threshold for what kind of ideal scenario it would be. Now, how does artificial intelligence work? Artificial intelligence works through a method, and this method we call it as a machine learning. This machine learning, it enables the machines to learn from the past data or experiences without being explicitly programmed. The same example I gave you in the case of Facebook or YouTube, where these portals are suggesting you the videos, and they are not suggesting you just the videos because they know you, but they know your history. So if somebody is watching your phone and suddenly some inappropriate content comes up and the YouTube is suggesting you, that means probably you might have seen some historically, that inappropriate content. And that's a reason, it is showing again and again, right? So that's how, it works there. Machine language works on algorithm, which learn by its own using historical data, and it works only for specific domains, such as if we are creating a machine learning model to detect a picture of dogs, we want this machine to see the picture and recognize the dog. So it will only give result for the dog image. But if we provide a new data, like if we provide a cat image to machine in front of the machine, then it will become unresponsive. It is not going to give you any result. So that's how it works there. Now, there are some subtypes of machine learning like supervised learning, unsupervised learning, reinforcement learning, and deep learning. So, supervised learning, you could when the machine is learning under your supervision or the supervision of the scientist. They determine, they are giving them the commands, they are feeding the commands, and they are supervising, what is the right way of getting the machine work done. So machine learn under the supervision. Unsupervised learning, where machine doesn't require any supervision and it is learning by its own experience. The more you feed, the better it becomes. For example, Alexa. When you bring Alexa first time in your home and you start talking to it, then it takes some time to get the results. But over the period of time, when you are repeating again and again the same command, daily at 9 o'clock, you are saying, "Hey, Alexa, what is the weather?" So Alexa is not going to work. Not going to take a time to tell you. It will immediately tell you because it has learned that, this is your pattern. So here it is the pattern recognition, but it is unsupervised because it's not fed or it's not under the monitor of any scientist or any other person, while it is getting the experience by its own. Then the third is the reinforcement learning. And in reinforcement learning, what is happening that, we are giving command. And when we are giving command, again and again, we are giving the same command, same command, same command. We are reinforcing the machine to learn the same thing. Okay. So this is a reinforcement learning. And then there is a deep learning. So deep learning, I will let you know, in details, what the deep learning is. But deep learning is the most complex among all the subtypes of the machine learning. So one term which comes and which is used in artificial intelligence is the artificial neural network. So when we, the biologists, they know that neural network is the connection or interconnection of the neurons. And this network system, artificial neural network, it is basically mimicking the same human central nervous system. Like our central nervous system, like brain and spinal cord, they are the most complex organs of the body. And they're the billions of neurons, they are connecting to each other, through different, the processes. In the same way, in the artificial intelligence, we make the circuits, we make the complexities, and we will try to, connect the different dots here, different data points there, the feeding points there, and we try to make it as complex as possible, and we try to mimic just like a human central nervous system. So here you can see here, so here it's the input data, and here is the hidden layer which does all the complex the complications or the calculations, complex calculations, and finally we get the output data. So now you can see this dot, it is connected with all 1 2 3 4 5 6 dots here. This is also connected with this is also connected with this. So that's creating a very high interconnectivity. In the same manner here also everything is connected with that. So they are creating a complex layer here, a complex system here, and they are making a artificial neural network. We call it as an artificial neural network. Simple artificial neural network, they will have one input layer. Here it is a one input layer, then one hidden layer, which does all the calculation, combinations, permutations, and then finally one output layer. So this is the simplest ANN. The more layer you start adding, the more complex calculations it can do, the more feeding you can make, and the more complex work you can get it done. So when we say deep learning is a subset of machine learning. While it has similar functions, but capabilities are different. It uses a layered structure of artificial neural network. So when we are saying less layers, we call it as a shallow. When we are saying more layers or we are introducing more layers here, we call it as a deep. So shallow and deep. So deep learning is when we are adding more and more complexity in this artificial neural network by putting more hidden layers, or by providing more input layers, or by providing more output layers, like, it making more complex. That is a deep learning. So the internal structure and number of layers within a neural network, it determines the ability to learn more complex tasks. Deep models, they have the capacity to process images, sound, and other high dimensionality data with good results, while other ML models may perform better with data organized into a spreadsheet like Excel part. So deep models are really making, impact in each and every area. And what I'm touching upon here is how artificial intelligence or deep models, they are helping the drug discovery process. There are some common deep learning architectures which are used in drug discovery. There are different types of architectures which can recognize patterns, which can extract high level features in distinct ways based on the structure of the training data. For example, one is the convolutional neural network. We call it as a CNN. It has a three types of layer which are commonly used. One is the convolutional layer, another is the pooling layer, and third is the full connection layer. So here you can see this is the convolutional layer here, and this is the convolutional layer. This is K and this is K minus 1. Then it can add K plus 1 also, K plus 2 also, plus 3, plus 4, and so on. So the more layers you keep adding, the more complex calculations it is supposed to do. So suppose this is an input image. Now we are getting only this picture and we are using only this area. So this area, it is coming in the convolutional layer 1. Other area is coming to the convolutional layer 1 at the other process. From this, it is going to the pooling layer, because all the pixels here which are represented by different convolutional layers, and based on the intensity, if it is lighter, it will be faded by here. But if it is darker, it might come here also. So based on these different convolutional layers, We also define the intensity of that particular picture. And then we pool everything here in the pooling layer. So all the data from the convolutional layer will be pooled here. Then we have the second convolutional layer, then we have the pooling layer, and finally we will have a full connection layer where all the data will come here and it will give us the output prediction. So this is a kind of convolutional neural network. Then we have the recurrent neural network. And as the name is saying, recurrent means that we are providing the feedback mechanism to that. This is especially aiming for handling sequence data, and it can remember the former inputs, whatever you have done, whatever you have uh, uh, fed into the system, it remembers that. So here you can see this is the input layer. Then we have the deep layer. We call it deep if it is more than one, right? And then we have the output layer. And whatever is coming here from input layer, the data is coming in the deep layer, and this is basically the hidden layers. And in the hidden layer, they are processing the data, and then they are sending to the second layer for further data analysis or for further data processing. And then based on this process, they have certain feedback which goes again to the first layer. And then again based on this feedback, they adjust their output and send it again to the second layer. So that's how it starts working. The more you give the feedback, the more better the results are, and finally they give you the output results. Then the third and the last is I would not say the last, but the important one is the generative deep neural network. This generative deep neural network, it is a powerful way of learning any kind of data distribution using unsupervised learning. So all types of models aim at learning the true data distribution of the training set so as to generate new data points with some variations. So we are providing here is we are providing them the already available data. And what we expect is that using the available set of data, they should generate some new data points with some variations as per our commands. But sometimes what happens, it is not possible to learn the exact distribution of our data. And so we try to model a distribution which is as similar as possible to the true data distribution. So that's how we work in this generative deep neural network. Now, two of the most commonly used and efficient approaches are variational autoencoders and generative adversarial networks. I will not go deep into them because it's not the presentation is not dedicated to the AI, but it's dedicated to the drug discovery applications. But it's important to understand the different architectures. So the function of autoencoder is to compress the data, and then the decoder function is to decompress the data and recover based on an algorithm with a minimal loss of information. While the GAN, it is used in image synthesis, image to image translation, and super resolution. So it is also used for the molecular structures, when we have the image of the molecular structures and when we are seeing in a reaction. So how using the image synthesis and then image to image translation and I think super resolution, and then we are seeing even a single bond change, it can be give the result and can get you a very new interpretation. Now, once you have this common architect, sorry, once you have this the deep neural network part, or you have the understanding about the artificial intelligence and how it works, I would like to introduce you some part about the ADME talks, or some part about how the drug discovery takes place. So drug discovery takes place with the target identification. We first identify the target, then we validate the target, whether it is the real target to produce the drug, or the new chemical entity which will going and attacking that particular or treating that particular thing. Then we identify the lead and we screen the lead. So we synthesize so many compounds. The lead compounds are identified and they are screened. Once we have identified and screened the right compounds, we optimize them. And once we optimize the lead, it goes into the preclinical phase. And then, it satisfies the conditions of the preclinical assays. Then it goes into the clinical. and finally it goes into the manufacturing. So from target identification to lead optimization, a part of the lead optimization, we call it as a discovery phase. So this is the drug discovery. And once we have the lead optimized, and then we are going to the clinical part, we call it as a development phase. And both phases are interconnected, and you can see it's a sequentially connected here. Now, when we say artificial intelligence in drug designing, the most important application is in the property prediction. The success of artificial intelligence for property prediction, it mainly depends on the data sets, the large data sets which are we have collected during the compound optimization for many different properties. And every big pharma company, they have a big data, or some data is also available in the public domain. So they use these big data set, and the more data we will have, the more chances of the accuracy and the right results we will have. So large data sets for targets and anti-targets, they are available across different chemical series, and they are systematically used for training machine learning models to drive the compound optimization. And many technologies have been used, for example, support vector machines, random forest, and Bayesian learning. So these are just the technologies. Don't worry if you don't understand all these things, but these are the name of the technologies which are used there. One of the examples is the prediction of the activities against different kinases. And kinases are very important class of enzymes, and which are useful in the drugs. So for the selective profiling in different kinase projects, they generate a large data sets, and they have been systematically used for model generation. QSAR and binary Bayesian QSAR models. In one of the experiments, they were generated from a large data matrix of around 130,000 compounds on 92 different kinases. And these models are applied to novel compounds to generate an affinity fingerprint, which is used to train models for the prediction of biological activity against new kinases with relatively few data points. So once we train our models with the existing compounds and with the existing data sets, then we can probably find out what is the biological activity against a new kinases. And here we are providing a few data points. So this is important here, that we gave the input of a very large number of data sets, but once our machine is trained to get the results, then we can provide only few data sets. We don't need to generate more and more data sets in order to evaluate the thing. And the machine can, based on its previous learning and previous training, can give you the results there. Then deep neural networks, they have been used in numerous property prediction problems. I hope by now you have understood what the deep neural networks are. So basically, when they have more hidden layers, they are the neural networks, deep neural networks. So here multiple nodes are interconnected, and the signals coming in from different nodes, they are transformed and cascaded to the neurons of the next layer. That's how we discussed in the earlier slide. So during training of a neural network, weights and biases at different nodes are adjusted. And since these DNNs, or the deep neural network, uses significantly large amount of hidden layers, a large number of parameters have to be fitted during the training of the neural network. These DNNs show comparable or better performance than other machine learning approaches. And some examples are, for example, different properties which are ranging from biological activity prediction or admit properties to physicochemical parameters here. Toxicity prediction is again one of the applications of the artificial intelligence. Here, one of the examples is the prediction of the drug-induced liver injury. So we used molecular descriptors like PADL, MOLD for molecular structure description, and heavy atoms, they are represented as a nodes and bonds as a edges. Then the graph is fed into a recursive neural network where the output of the next element depends on the previous element here. So by using this approach, 475 compounds were tested, and the prediction accuracy was 87%, sensitivity was 82.5%, and specificity was 92.5%. And that's how, within using the machine, I think, this achievement was pretty uh, good and significant. So deep learning has also been used to predict potential energies of small organic molecules and replacing a computational demand quantum chemical calculations by fast machine learning methods. So if we apply all these things in drug designing, probably, we can shorten the time of the discovery. Now, another applications of artificial intelligence in drug designing is the de novo design. And de novo design means when it aims to generate new active molecules without a reference compound. And because this is very tough and it's a very, very time and dollar consuming process, so it is not common in drug discovery. So one approach is the variational autoencoder, and the variational autoencoder consists of two neural networks. One is the encoder network and other is the decoder network. So the encoder network translates the chemical structure into a real value continuous vector and as a latent space. And the decoder part, it is capable to translate the vector from the latent space into the chemical structure. So what you can see here is this is the structure of a chemical where the encoder compressed it, put it into the latent space, and the decoder network, it extracted the information and reproduced the same molecule outside, giving the results. So what is the benefit here is that by compression of the data using even a small models, you can feed, you know, large large number of molecules here, and then you can get the calculation done. Now, another application is the synthesis planning, because ultimately, you have to synthesize the molecule. You have achieved what you want, you have designed the molecule against a target, and now you have to plan the synthesis. So, in certain situation, synthetic challenges, they restrict the available chemical space which is accessible for design molecules. So, synthesis planning is a very key discipline in drug discovery. There are two types of planning. One is the forward synthetic planning and other is the reverse synthetic planning. In forward planning, the DNN has been trained with a set of millions of reactions and it outperforms an expert system for comparison. So here you are giving the direction in the forward reason where you are putting a different millions of reactions feeding into the system, and then you are asking which is the best one, and it is with the comparison it is giving you the result. In retrosynthetic or reverse planning, reactants and products are coded by the software SMILES strings for recurrent neural networks, and they are coupled to each other in the encoder decoder architecture, and then it is giving you the result. So in both ways you can use and you can plan your synthesis. The combination of the three deep neural networks with a Monte Carlo tree search for retrosynthetic prediction, it has yielded an excellent performance. So people have used this and now they have published the results, and they have got a very good the encouraging results out of their experiment when they have applied the deep neural networks and using a Monte Carlo tree search. So once we have the molecule, we call it as a NCE, which is the new chemical entity. We move to the ADME-tox studies, right? Now, people who are not familiar with ADME-toxicity, admitoxicity or admitox is the disposition of a pharmaceutical compound within an organism. So A is the absorption. So we see how a drug is absorbed into the blood stream via the intestine. D is the distribution here. We see how a drug is transported into the blood to its effector site. It reaches to tissues and it reaches to what organs there, how much time it takes to reach there. M is the metabolism. We see how a drug is metabolized in the body, what is the mechanism, what is the route, and what are the metabolic effects. Most of the metabolism takes place in liver, but other organs are also involved in metabolism. So, majority of the drugs or metabolic activities are used or are done with the help of the liver enzymes. Then excretion. E stands for excretion. So how a drug or metabolite is removed from the body via excretion. And finally, we do the toxicity studies, whether a drug or metabolite exerts a harmful effect on the body or not. And this toxicity study is very, very important. If you don't do this study, or you miss this study, or you miss the data, then after spending millions of dollars on a particular molecule, you have to retract the drug, and ultimately it is going to be a failed venture. So admitox is the ADMET: absorption, distribution, metabolism, excretion, and toxicity. Now, why is admitox important? Because the FDA says the preclinical requirement is there to evaluate a drug candidate for DMPK toxicity and efficacy effects. Both in vitro and in vivo laboratory testing is needed, and without this data, FDA is not going to give you any approval. ADME assay is typically used in the discovery or lead optimization. So they do a basic research or screening of many compounds. Many labs do. They extract the natural compounds, and then they see whether the particular compound is effective. They use the in vitro models, they use the cells, they use the various assays, advanced assays, and in development, where they characterize the properties of few compounds which will be submitted to regulatory authorities. Mainly, then the development part is done by the pharma companies, because uh, they have to synthesize and they have to market the particular product there. Usually, from target identification to the manufacturing, the time period is more than 10 years. On an average, it is 10 to 15 years it takes, and the investment is billions of dollars. So there are only few companies which have the capability to do this whole process, the whole complete things. But there are small companies also which come and they do the assays, like development assays, they do lead optimization, preclinical, clinical. We call them as a CRS, or contract research services, or contract research agencies, or contract research industries. They do this development. But ultimately, this target identification to manufacturing, it is a very, very costly affair. Now, we see how artificial intelligence can be used in admitox. In the early stages of drug discovery, a perfect in silico in vitro correlation is not at all required. If you have some agreement with the data, you can move forward, no problem. These models, they should provide better understanding to enhance the optimization process. But in the later stage of drug discovery, the bar slightly goes up. And then, the models must differentiate molecules with a very small structural change. And any small structural change is a new molecule. And then it has to start with, again, the beginning. And in the later stage of optimization, a local model may outperform a global model. So we don't need a universal model to come into the picture, while a local model based on our own requirement, based on the molecule, based on the application, it will work. So, in other words, the admitox machine learning model used at each stage should be fit for purpose. It is not that that at one stage you are using model A, in other stage also you are using model A, in the third stage also you are using model A. It is not going to work there. Every stage has its own need. Every stage has its own applications. Every stage has its own way to interpret the data and submit the data. So we need to have the right model which can just give us the purposeful results for that particular stage. These are some of the examples where the admitox properties are modeled using machine learning algorithms from the in vitro or in vivo data. So, for example, the solubility, we measure the solubility by measuring the kinetic solubility. We see the clearance in rodents or small animal models, and it gives us a snapshot of the pharmacokinetic properties of the drug. Then we do permeability assays either using PAMPA or using CACO2 or MDCK. So PAMPA is a passive method where we simply use a filter membrane based wells, and passively we see how much drug has been transported from the upper compartment to lower compartment, or lower compartment to upper compartment in a defined period. While CACO2 and MDCK, these are the cell lines. This is the canine kidney cell line, and CACO2 is a human intestinal cell line, where you grow the cells on the inserts, or the we call them as the inserts, where you have a well-like structure, and then you have a membrane. These CACO2 cells, they cover up the whole membrane, where you put your compound on the upper compartment, and after a certain time you see how much has reached to the lower compartment, crossing the layer of these cells. So this is a monolayer culture, and MDCK is also a monolayer culture, and this gives you a good idea about the active transportation also, because these cells, they have the transporters uh, which are expressed on the membrane. Then we have the transporters assays, like for example, the PGP, and which are efflux transporters and some other influx transporters. And we use the overexpressing cell lines, and we target a particular transporter, and we try to determine, our new chemical entity, how it is entering into the cell, which transporters are used, or whether it is modifying those transporters or not. Then we do the metabolic stability assays using the liver microsomes and hepatocytes. And liver microsomes, as I told you, because most of the drugs are metabolized in liver, and we use these liver microsomes and the hepatocytes models to check whether our compound can be metabolized, what are the enzymes responsible for the metabolism, how early or how late it can metabolize, what is the stability of this metabolism. Everything we need to check, and we need to produce the data. Then drug-drug interactions. We use the CYP450s and transporters. So CYP450, again, it's a class of the enzymes which metabolize all the xenotic compounds, and transporters, they are used to you know I allow the drug to go inside, or just to flush outside. So we use the drug-drug interaction studies to see whether the existing drugs are competing with the new compound, or whether they are producing some toxic effect, or it is really harmful to the body. So we do all these studies using drug-drug interactions. Then blood-brain barrier, which is really very difficult to study. We use the mouse brain endothelial cell line. It is one of the examples where we grow the cells, and we try to see whether it can cross the barrier or not. Because if we want to target the brain diseases, we need the drug to cross the blood-brain barrier and go into the brain, and that's the target there. Then cardiotoxicity assays or hug assays, we use the binding or flux on different type of cell types. So these are all the measurements and the properties which are defined by these measurements. They have been modeled using the machine learning algorithms to help us into getting more precise in vitro or in vivo data, and to help us to shorten our time, because time is very, very important in any drug discovery process. These are some of the examples of the machine learning algorithms which have been applied in AdmiTalks, like random forest. Random forest is a method. It constructs mainly decision trees and outputs class or mean prediction. Then we have support vector machine, which is again a supervised learning method, and the examples are mapped in space and classes separated by hyperplane, and then we see them. I think I don't want to go into deep, because I know that it's something not required at this point of time. But the objective of showing you this slide is that these are the algorithms which have been used in the AdmiTalks studies. So the neural network, k-nearest neighbors, naive biases, and deep learning models here. There are several companies that are using artificial intelligence for drug discovery or the aspects of drug development. And these companies are like, I have listed here some companies. If you wish, you can just go to their website, and you can find more information about how they are using. Probably most of the data or most of the things are proprietary in nature. They might not give you a trying, but in case if our students want, they can apply their internship in these companies, and then probably they can have a good hand experience here. So I'm just going to give you two, three points here in ethical dimensions of artificial intelligence. There are few questions which we need to answer here. Is like, how and to what extent the algorithm processing contributed to the decision making when we are making a drug. Definitely tomorrow when we will say that FDA, we will go to FDA, and FDA will definitely ask us that you have used the machine learning language, or sorry, machine learning, you are the deep learning. So, or using the algorithmic processes, what is the percentage of contribution? And if they find it that, and you say that, okay, I have used a significant portion or significant, percentage in my whole process, then there are questions, further questions about that. How do you know that the language, the learning you have done, or the results you have got because of this uh, uh, artificial intelligence, they are pretty good, they are really true, or they are not influenced by your wrong inputs, they are not influenced by, the wrong essays or the wrong data which you have fed. So you have to be very ready about answering all these questions, because ultimately the FDA will uh, give you the approval only when it is satisfied with all the answers you are providing and with all the evidences you are providing in your report. Then which data was processed and what is the source, you need to know, because when they go to the reverse investigation, they will go up to the source, and from the source they will try to investigate. Or in case if your drug is in the market, and if something goes wrong with the drug, you have to no other option but to retract the drug from the market. Then definitely FDA is going to investigate the whole process, and then they will be requiring all the information. So we need to be very, very careful when we are using the artificial intelligence in our assays. Then how parameters were treated and how they are weighted, and whether they are treated and weighted in the SOPs and the way it should have been done. So everything has to be, put into the picture. So I am going to simply, finish my talk here with certain takeaways here. Uh, First takeaway is that the models can be used to guide design prior to synthesis or narrow down the number of compounds. So that's very, very helpful, and that's the reason the AI was important step in the prediction of the properties. Then the size and quality of data sets, they are the prerequisite for enhancing accuracy of the result. It is like, what is, you must have heard about the GIGO, garbage in, garbage out, right? So whatever you are feeding into the, it's a machine, whatever you are feeding, it is giving you the result based on that particular data sets. So it's very important that the size should be large enough and the quality of the data set should be highest, so that the results you will get is the most accurate one. And then not all algorithms can be applied universally. There has to be a tailor-made approach, and this tailor-made approach has to be done whenever you are planning an experiment, or when you are taking help of the artificial intelligence in your experiments. So with this, I would like to thank you, and if there are some questions, I would be happy to take.
So, hello, Bhuti sir, are you there or should I take on? Hello.
Yes, I am here.
Sir, the talk over, and please take on the session forward. I would request you.
Thank you, sir.
Thank you, sir.
Hello?
Sir, we are
Hello?
hearing.
Yes. So I think there are some questions at my end from YouTube. I think sir can see some of the questions from his end if possible, or maybe he can just open it for a discussion.
So, I have not received any question.
So here I can see in the chat box there are questions. So probably So, with your permission, we would like to ask on behalf of students.
Please help Bismita madam. बोल
Yes.
दीजिए.
Okay, sir. So actually, there's a question from YouTube that how can a pre-designed artificial intelligence system for a drug can be used for repository in one drug scenario and can be applied to other diseases? Like, is it possible to apply the same artificial intelligence for multiple disease purposes, or what should be the technicalities to modulate that kind of thing?
Okay, so as I discussed in my presentation, that when we are designing a particular drug, we are designing a drug against a particular target. So when we are seeing the multi-models here, and as per the questions, if I correctly understand, is they want to ask whether the same model can be applied to different diseases, or right? So is it the thing, Dr. Bismita?
Yes.
Yeah.
It's the same.
So right. So it is. I would say the answer is no, because when we are targeting a particular disease, the disease is different, and the parameters are completely different. So when we are designing a particular uh, new molecule using the artificial intelligence, the data which we are feeding, this data should cover the aspects specific to a particular disease or to a particular target. And since different diseases have different targets, even a single disease may have different targets. It's not that the different diseases will have different targets. If we are, for example, if we are looking at uh, the let's say an example of the diabetes. So it's not about that the just the insulin is the answer to diabetes, but there are certain medicines also which help to lower the blood sugar, and they have the different ways. If we are targeting the hypertension, there are many classes of drugs which are available and which act upon the different targets. Some work upon the different enzymes, some work upon the different receptors. So the targets are different. And since the targets are different, although the outcome is the same, we want the desired outcome is that it should give the result the same, but the targets are different. So we have to be very, very careful when we are using the artificial intelligence models, and we one fit of all approach will not work here.
So actually, there is another question from Prabhat Kumar Swami. He is asking, how can we consider big data is a safe and secured method and system and not be used?
Well, well, that depends on where what is the source of the data, and that's the reason it is a very complicated process. It is the reason the artificial intelligence is not that much easy, because there is no way to know whether the data is really right or wrong. But the data which we are getting, for example, there are so many public domains. There are so many portals which the researchers, they on their own wish they submit the data, right, whatever they get, and this all data is pooled, and then this data is given to the public and open to the public, so that public can use it and they can analyze it. Now, some data is confidential, some data is only generated by the big companies. And these big companies, when they are generating the data, they use it for their own purpose. They never put it into the general domain. The question is whether the data is true or data is false. It all depends upon, how honest is the person who is putting the data into the system. It is just like that, we are publishing the paper. And when we are publishing the paper, we are putting some data here, and there are so many instances where the papers are retracted because they either modify the data, or they engineer the data, or they do that. So these kind of things are there, and when the data, so it is very, very difficult to say whether the data is right or wrong. But yes, if it is I mean if it is generated by some companies and they are the proprietary, usually it is very safe and very good data. In public domain, I would not say that the data is not true, but I am saying that it is very difficult to, unless and until the person who is submitting, he has a reputation.
So nowadays, you might be knowing that there are many apps generally. So before you open and use it, they are asking for your location, your private data. But unless and until you say yes and allow them to share your data, your photographs or many things, they are not going to allow you to use the app itself.
Yes.
So in the background itself, that's why I think there are many apps were banned, and due to there are some reasons for international national security and all.
Yes.
So how can, do you have any checkpoints where we can just deny to give the, but if we do, then we are not able to use the apps. So is there any thing where we can restrict our information to be used by other people?
Well, in this case, it is very simple. If the app is asking for the data which you don't want to share, it is simply you don't, keep the app in your phone, because there is no other way. We cannot say and we cannot fight with them that we will not give the data, but we will use the app, because the app is, made in such a way that either it is yes or it is no. If it is yes, you are opening the whole thing to them. If it is no, it is no. So if such kind of scenario comes, it is very, very simple. Say no to them.
So there is one more question. Like, is it possible to invent some nano robot in the near future, which will present in our body and repair our defected cells? Is that
Ye-
something possible?
Well, first of all, there are two aspects of this question. First is the making of a nano robot, and second thing is repairing of the cells. So let us, I'll answer first question is that, yes, it is definitely possible. People are working on it. There are so many things. They are making a molecular machines, and that molecular machines are giving us hope that one day probably they will, take out all the clogs from our arteries, or they will do some other things. So answer is yes. Now second thing is repairing of the cell. That comes, further, with that. So let us evolve. Let us come up with some prototypes, and then I'm saying, future, you cannot say no to anything. Anything can happen. So that's how.
So last, I'll just take on one, that is uh, the AI drugs, the artificial intelligence drug design. Do you think it's better than conventional techniques, and how we can put ahead?
Sorry, which drugs? First part I missed.
So AI-based drugs or
AI
artificially
based drug.
intelligence
Yes,
drugs, yes.
yes.
So
So
how they are better than conventional? What is that one or two aspects which we add, and we can say that yes, definitely for these reasons only, these are, we can consider them as better than conventional ones.
well, I would put like this, that earlier, in our earlier generations, people used to calculate on page, using a pen and paper.
Yes.
Then the calculator came into the picture, and this calculator helped to shorten the time and put the calculations much faster. Then the computer came, and this computer made the things pretty easy for us. So the same thing is here for the drug discovery also. When we are using the artificial intelligence only helping us in our essays, helping us to do, it's not replacing the human being, it's not replacing the actual essays, but it is actually helping us to narrow down the things, narrow down the molecules. So the thing initially, which used to take around 5 years for a new molecule to come to the stage where we can do assays, probably with the help of the artificial intelligence, it may take maybe a year or maybe 6 months. But that's in the near future. So here, what we are talking about is that we are making use of artificial intelligence to help our drug discovery rather than giving complete dependence on the artificial intelligence for the drug discovery.
I think last but not least, I have a question, that is why it is difficult to model endpoints such as the time-dependent CYPs and inhibition, like those kind of systems.
Right, because the reason is very simple. When we are saying the time dependent inhibitions, the things which come into the picture is the three-dimensional aspect structure of the molecule, and the fragments, the structural fragments which come into the picture. So when these things come into the picture at this present point of time, we have not got success into mapping all these things into them, and that's the reason it is difficult. One of the example I would like to give is the mitochondrial toxicity. So in the mitochondrial toxicity, there are so many mechanisms which can lead to mitochondrial toxicity. Now, which mechanism can act upon, or is induced by a particular drug, it is not possible to predict with the AI unless and until you have all the mechanisms available here. So that's the reason this is lagging behind.
think we have already taken all the questions, and there are a few more questions, but probably we'll take on further.
I think there
Can
is
I
one more
ask a
question.
question?
Yes.
Mrityunjay, are you there?
Yes, ma'am.
Yes, okay.
Yes, ma'am.
Okay.
Okay, Dr. Verma, it was a very good session, and a lot of technicalities was involved, and we really liked it.
Thank you.
Sir, I just have one question. Like in previous month, this drug discovery hackathon, we participated in it. This is conducted by government of India.
Sorry, your
So,
voice is cracking.
Mrityunjay, can you come again with your question?
Yes.
okay, is it now audible?
Yes.
Yes.
Okay, so previous month actually we participated in the drug discovery hackathon that the government of India is actually conducting. So in that hackathon, we had used many techniques to find the drugs that is available that might be potential in inhibiting the SARS-CoV-2. So in that we had used this many ADME softwares. Means there are many ADME softwares which is present online. So can you suggest any specific because which is much more efficient than the ones that we used? We used Swiss ADME for case. So is there anything which is better than Swiss ADME or any public domain kind of softwares?
Well, you give me some time. I will find a good software for you. But at this point of time, difficult for me to
There are
give you
many
an answer
softwares,
because
and
there are so many.
most,
Yes,
yes.
there are so many
So there
softwares.
we cannot say actually that
Yes.
this is bad, this is good.
Yes.
There might be some
No.
good technicalities in one aspect, there might be some good technicalities in other aspects.
Yes.
So depending on your requirement, it actually works. So either you go through many of the things, you look for what you need to do, and probably the softwares can help. There are many softwares, I can tell you from my experience that not all the softwares are equally good for the things you are looking for. So what exactly you are looking for, if you are sure enough,
Actually,
probably you can come and we can.
Lipinski, yes, ma'am. Actually, this Lipinski rule of five that they have for every small molecule. So that is exactly what we were actually finding. Yes, sir. So thank you,
Yeah.
sir.
So I think later on also we can connect and discuss the problems if you have, and we'll see together,
Definitely,
find out some common paths where
Definitely.
you can proceed through your progress through your project. Okay. So I think Sujit Bhutia sir will take on. Sujit Bhutia sir, are you there? Bhutia sir. Hello. Hello. Yes, sir.
Yes, ma'am.
Sir, with Dr. Vipin Verma. So,
Thank you, Verma sir. Very nice talk.
Sir, thank you so much. And I wish everyone is stay safe in this environment. And thank you very much.
Be safe and be happy, sir, in home.
Thank you, sir. Same to you, sir.
sir,
So,
Sujit sir.
our
Sujit
next
sir.
speaker is.
Sujit
Yes,
sir, hold on. If with your permission, I think Dr. Veena is here. In the morning session, there was some technical error, so that's why just her internet connection went off. So with your permission, at least two questions we would like to take on with Dr. Veena. At least, so she's here. Dr. Veena, are you here? Probably.
Yes. Yes. I'm so sorry about today morning's snafu from my end. I'm here now. Dr. Bismita requested me to join for a short while and take some few questions. So I'll be more than happy to answer them if I can, if I know the answers, and if I don't, I'll get back to you with the answers. If you give me some time.
So before Dr. Venkat's session, I had requested him to wait. I'm putting him into hold, probably with permission from Dr. Bhutia. So just quick questions. What are the safety measures have to be taken during manufacturing process, and the various kinds of controlling factors which we can actually, for safety measures, we can take control while the initial processing part itself is there. So major things if we can control in the initial processing and formulation part.
Correct. So actually, the guidelines for safety is very much safety. So I understand the safety of the sterility of the product. Did I get that correct? Safety.
Yes, safety
Okay.
and the process and everything.
Correct. So in that case, you have to, so once you start the manufacturing process, it's very essential where we follow all the GMP requirements. And the GMP requirements includes the infrastructure. You have specific requirements as to what should be. There should be a clean area. If you're doing a fermentation, designated areas, what are the specifications that are required to be met in those areas? All those have been specified in detail by the regulatory agencies, especially the process of GMP production. That is good manufacturing practices. And it remains the same for to a larger extent, it remains the same for small molecules as well as for the biologicals. But in addition to that, if you are looking at safety from, say, if you have developed a clone and you're looking at the clone development, you have a cell line that you have identified, you have made a permanent or a stable cell line that expresses the protein of interest, then you have to make sure that with every manufacturing process, every time you seed it and you grow up the cells, you're sort of introducing any variations or mutants in those cells. And you have to use the high-end genomic technologies to characterize the cell lines, and you have to furnish all the data to the FDA. So that is did I answer that question correctly, or is there?
Yes, mostly it's understandable that we need to focus on those areas, and we should control all the parameters for cleanness.
Correct. The same. There is many more. Sure. There is many more parameters, even if you're looking at batch-wise fermentations or you're moving into the purification process. So each of this is very tightly regulated, and you have to have a very good quality management system already existing in the company. Because as you know, the whole principle of manufacturing is quality by design. And at each step of your process, you have to ensure quality. It is not something that needs to be looked at the end of the product, at the end of the process. So each and every process, even if you are just looking at your clone development, you make sure that the best clone gets into the seed culture. If there is a seed culture, you look at all the processes, dissolved oxygen, what is the pH? If you're going to grow it for 3 to 4 hours in the seed culture, you make sure that the ODs are right. And you have a very good quality management system. Make sure that all the assays that you're using to evaluate the quality are validated assays. So it's a concerted and a well programmed process. So it's quite intense, and it requires a lot of stakeholders from different fields to come in and to ensure that at the end of once you take out and seed the culture and you get the final product which is ready for fill and finish, you really have the best product in going into the market or going in for testing.
So I think we had a lot of questions, but one fine question I would like to take on with you. So while preparing from batch to batch, we call it a lot culture, from lot to lot, that thing. So how from one process, like if we, suppose I'm using a commercialized dye or a protein, and if I have a long-term experiment and I'm using it in various kinds of experimental setups, then how I can make sure that the lots or the prepared processes, how they are similar to each other. Like they might not be equal, like coming a chemical substance or chemicals coming from one lot may not actually give similar kind of, they have similar kind of nature, exactly similar, and how we can actually correlate among batch to batch.
Right. So, right. Actually, that's a very good question. The thing is that it all depends upon the final use of your product. So if you're planning to use it, in vitro as an in vitro reagent, then probably the amount of stringency in the quality is much less. But on the other hand, if the protein is going to go in as or going to be used for human use or medicinal use, then it has very high levels of quality that it needs to be met. For example, every product that goes in, it is either will be pharmacopoeial in nature. So say, for example, you're talking about insulin. Then insulin, there is an insulin monograph that is available from, you have the USP, that is the United States Pharmacopoeia, or you have the Indian Pharmacopoeia. And these pharmacopoeias give us a monograph. That means once the product is released, or you're talking, either you're talking of the drug substance or the drug product, it has certain assays, certain qualities it has to be met, and how that quality is evaluated. Say, for example, if you're looking at the assay, so you are going to do some assay to measure what is the amount of insulin in it, which would be probably HPLC method or the size exclusion method. Now, when you prepare insulin and you manufacture insulin, you will have a lot of dimers and a lot of other, impurities that come into the process. Okay, while you are purifying it. So there are certain specifications that are required for the final product to be met. Like you will have the host cell protein should be less than some specification, or you will have the endotoxin level should be less than 1000 ppm or maybe 100 ppm. So that's the monograph. So the final product that comes out has to undergo a assay. So in the end, what happens is after it meets all the requirements and specifications, in the end, what the QC people release is something called as a certificate of analysis. You might have seen that. Actually, even when you buy reagents, you would have seen something called as a certificate of analysis. That's the final quality report that comes out. You have to ensure that all the specifications are met. When you are talking about biotechnological products or biomolecules, it's very difficult to actually get the exact product out every time. Because like you had rightly pointed out. But what you can do is bring it as close to each other, keep the variability to the minimum. And you can do that by sticking to the monograph standards, which this is already there. Like you can always refer to the IP if it is in India, or the USP, or the European Pharmacopoeia, or you could also refer to the Japanese, because they are the leaders in this. So they already have certain standards already set for the product, especially if it is for the medicinal use or the human use. Hello?
Yes, ma'am. Thank you.
Yeah.
Okay. I fully
Okay.
understood. Hope students might have got your the answer. Hope they understand. I always, in my classes also, I always emphasize on making everything to be on a same platform, getting
Correct.
into the same.
Absolutely.
So
In fact, it's
something is
sorry. No, I don't want to belabor the point, because I don't want Venkat's talk to be extended. But I just wanted to say that it's so important that we do the right documentations, and also the methods, sticking to the methods. You have an SOP, you have to stick to the SOP. That's the only way you can reduce the the variations in the final product.
Okay. Thank you so much.
Okay.
Thank you, Meena.
Thank you.
Thank you so much.
Thank you so much, Dr. Vismita.
Yeah. So, Dr. Sujit, hello. Dr. Sujit, Udaya sir.
So we can start, madam?
Yes, definitely. Yes, we are delayed, but...
So we are sorry for the delay. I welcome Venkat Raman, who earned his Master of Science in Biochemistry at Bangalore University, Bangalore. He spent 15 years in industry, engaged in manufacturing and development of molecular biology product. He held, he held roles in R&D, manufacturing, operation, and management. In 2020, he earned his executive management program at the Indian Institute of Management, Bangalore. He has vast experience in development and commercialization of diagnostic assay. He worked at premier institutes in India like Amrita Institute of Medical Sciences, and having international work exposure at various commercial organizations located in Netherlands, Spain, France, Kuwait, and Dubai. Venkat joined at Biomerit in 2016 and currently manages in R&D section for molecular diagnosis portfolios. He is dedicated in providing solutions in molecular diagnosis assay. Let's listen his topic, molecular methods for diagnosis and characterization of previously uncharacterized pathogens. So I welcome Mr. Venkat. Please, sir.
Thank you so much, Dr. Bhutia, and thank you so much for the Department of Life Sciences, NIT, for giving me an opportunity this afternoon to have an interaction with the academicians. Being in industry, it's been always pleasure talking to academicians and the students to learn the current research, what is going on, and cope up with that, and convert them into the industry research products. Actually, today, hopefully, the talk is more relevant of current scenario in which we are. I'll take up that without further delay. Let me share my screen. I'll start. Is it visible, my screen, Dr. Bismita?
Yes, yes.
Okay, thank you. So today's topic, I wanted to cover the molecular methods for the diagnosis and characterization of previously uncharacterized pathogen. So the title is so vast in terms of few aspects. Before going to the outline, I'll tell you it's very difficult to cover the entire set of molecular methods which we would be using are available for the diagnosis of characterization of pathogens. And if you are dealing with uncharacterized pathogens, if the pathogens belongs to virus group, or if the pathogens belongs to bacterial group or fungal group, so again the set of molecular methods which you need to use it for characterizing them and diagnosing them is different. So what I would do is I would start my outline of the webinar with an introduction where you would find what are the different molecular methods which are available for the diagnosis and characterization of novel pathogens. And then I would come to the topic of molecular diagnostic kit development from an industrial point of view. It is where many academicians or people wanted to know what actually happens at the industry level, how the industry level diagnostic assays would be developed, and how they come into the picture after once the commercialization aspect has been done. And I will try to summarize that what is the way that going forward, if you come across another pandemic kind of situation where we are in today, how to deal with them, what are the type of methods which you would like to use it to deal with those kind of pandemics and give the solutions as quickly as possible so that the humankind may not suffer as we are till today suffering actually. So without the further delay, I'll start with an introduction. In this topic, before going to the introduction part, I would like to mention a disclaimer that I'm working from RAS Life Sciences. It's a bioMerio company. So none of these contents which I would be talking and also showing in the entire talk, neither my company is formally supporting them or would recommend them. It is purely a personal level talk. So whatever the content and the presentation has to be taken with the note that it is purely from my end. So I'll start with an introduction. Before introduction, I would like to mention that what is an outbreak, epidemic, and pandemics? Why the reason that I wanted to bring the definitions of it for your notice is, as we all know that today we are in a situation where we are still suffering with the COVID-19, which is a perfect example for the topic. When the humankind been challenged such kind of pandemics, what is happening is the economies of the world is tumbling and the humankind is suffering, the life is getting halted, so many situations which is adversely affecting the human life is getting open up whenever these kind of situations are coming up. But then being scientists and in the research, how we could able to support the entire humankind with the solutions is the theme of the topic, which we would take it forward with an simple examples and the with the simple definitions. I would like to start this. Outbreak is nothing but a sudden rise in number of cases of a disease. You could simply put it in that way. That means perfect example for that would be in influenza, that sometimes you get in a cold season, you get a bunch of people suffering with cold, etc. So it would be called as an outbreak in a particular area, kind of like that. And then what is an epidemic? Epidemic is can be put in simple way that it affects a large population in a particular community or a region. Example could be for those kind of epidemics is SARS. So it happened to be later pandemic also, but then it largely pertaining to the epidemic kind of situation where large people had suffered with the kind of with these kind of infections. And then what is a pandemic? Pandemic is also an epidemic, but it is spreaded over multiple countries and continents. Today, if you are talking that, then the perfect example for this pandemic is the COVID-19, which is also under a category and been announced by the WHO as a pandemic situation that COVID-19 is. So it has been already spreaded over 130 countries and millions of people are getting suffered. So this is the perfect example of a pandemic actually. So I have a next slide which is showing you the history of pandemics which humankind had been suffered or been challenged by them. I took very recent century pandemics which had happened from right in 19th century and the recent ones. So this slide I have shown up in 1918 we had a Spanish flu which led about half a 0.5 billion deaths actually. And then we had 1957 Asian flu, which led to 1.1 million deaths. We had 1981 HIV, which closely 35 million deaths and still going on. And then we had 2003. At 2003 we had SARS. Maybe probably this is the least in terms of deaths which had been occurred. About 1,000 deaths had occurred so far with SARS. And the current one, COVID, which had occurred early in December or November of 2019. COVID-19, it's already led to the death of 1.5 million. So what does this data shows? I mean, this data primarily shows us two things, at least if you notice that every 20-30 years, humankind is getting challenged by these kind of pandemics, which would eventually led by some bacteria or virus or some of the pathogens which is transmitted from, escaped the transmission cycle from one organism to the another species or organisms and then causing the disease and alerting the humankind to subsidize the infection and act upon, asking the government to act upon those kind of pandemics. So, but then what is happening actually in terms of readiness, our readiness, or in terms of the methods available? Where are we ready? When if once asked that question, actually, the current situation of COVID-19 was a perfect example where where we had, as a human race, substantially shown that in terms of vaccine development, whether it could be a drug discovery, whether it could be a diagnostic kit development, we had eventually shown that yes, it is possible, unlike the traditional ways that we take years in terms of development of any of these, whether it could be a drug for the treatment of infection, whether it could be a vaccine for the immunization of the entire human race, or whether it could be a diagnostic kit to stop the infection or contain the infection. So it is possible with no time. This is what been shown as, and I'm very glad that we are in a situation now that these kind of pandemics further in future also will can be tackled very nicely. So with that, I would like to tell that traditionally there are ways that you usually characterize the pathogens by using uh, your culture methods and your serology methods, and the including the biochemical methods which are available in labs and for the research. But the molecular methods over recent years had shown prominent and pivotal recognition in terms of as a set of tools available for the researchers, that it could be rapid and it could be cost effective to deal with an identification of uncharacterized pathogens. So my entire topic or talk would be around of uncharacterized pathogens, especially on viruses. The reason is that when you are talking about again uncharacterized pathogens of a bacteria, it would have to you would have to deal with a different set of molecular methods actually. That's the reason I'm limiting my talk only for the virus discovery. So please consider that.
So, Dr. Venkat, can you just make the PowerPoint full screen if you have options?
Yes, please. Wait. Yeah, is that now fine?
Yes. Thank you.
Okay, good.
Okay.
Thank you so
Yeah.
much. Yeah. So I just wanted to confirm you that the most of the talk would be on uncharacterized pathogen, especially for the viruses. Here in this slide, I have tabulated or formed sentences which are very relevant, that how molecular methods are superior or helping along with the traditional methods in terms of discovery of novel pathogens. Actually, there are molecular methods which in combination with the traditional methods, or in comparative to the traditional methods, they could be cost effective, because some of those methods which I would be discussing in my talk will definitely show you that how cost effective they could be when you are dealing with certain kind of surveillance studies or pandemic situations. The discovery could be, and after post discovery, that to develop the diagnostic kits, how the cost of the diagnostics is considerably reducing by the molecular methods that you would see. And they are also specific comparative to the traditional methods, such as serology methods, because molecular methods usually detects the pathogen, they don't detect the immune response, they don't capture the immune response, which means that it is more chance that you are ending up specifically detecting a pathogen instead of getting the immune response or a kind of biochemical response which you would see across a family of pathogens actually. And they are also rapid compared to the traditional methods. The reason is that many of these methods which I would be discussing in my topic shows promising turnover times with no time, like in case of happened to be COVID-19, that the discovery of pathogen happens in months time. wherein in others case where few culture not possible organism or the organisms which are slow growing, in those cases it would be difficult and take a lot of time for their discovery. So that had been avoided, that can be avoided by choosing the molecular methods. So as I mentioned before, there are various molecular methods are available for the pathogen characterization and diagnosis. It is a vast list from which I have chosen selectively few splendid molecular methods which would be which had been extensively used in virus discovery and virus discovery. One of such is PCR, polymerase chain reaction, especially family-wide PCR method, which is a form of polymerase chain reaction. I would discuss a little more in detail in my upcoming slides of this. And then you see that how this approach or method actually helping to deal with pandemic kind of situations, or in general, if you come across, if you wanted to do any metagenomic studies, or if you are doing any new pathogen discovery, how this approach is helping you. And then real-time PCR. Real-time PCR is another form of PCR, polymerase chain reaction, wherein quantification is possible through hybridization and fluorescence-based methods, actually. And then you have DNA microarray. DNA microarrays are very vastly used in novel virus discovery and bacterial discovery as well, actually. So DNA microarray works on a principle of hybridization and then fluorescent emission, actually. So even this technique or method is quite widely used in discovery of novel pathogens. And then we have last but not least one, the nucleotide sequencing. Nucleotide sequencing has changed the entire world or molecular biology, revolutionized the molecular biology in terms of looking at the prospect and the discovery of pathogens, actually. That with its quick turnover times and with its the enormous amount of data generation, which I would be talking a bit later, the entire, I mean, it had become now a readily available and very vastly used tool in viral discovery. So in this slide, I would be discussing the workflow. When we are talking about the molecular method workflow, I could generalize the common workflow of a molecular method. It always starts with the sample preparation. So either your sample could be, I mean, when I mention sample, it could be a specimen of blood, serum, plasma, frozen tissue, CSF, nasopharyngeal specimen, culture cells, stool, or urine. Where any one of these which are the pathogen can be your sample, and the molecular method workflow starts with that. You would be preparing the specimen, decontaminating the specimen, and then you would be extracting the RNA or DNA, which is a nucleic acid of the pathogen, and then you would be putting them on amplification in the second step. So in amplification, either you would be using techniques such as PCR, real-time PCR, reverse transcriptase PCR, reverse transcriptase real-time PCR, isothermal assays such as LAMP or other techniques. And also you would be using sequence independent single primer amplification techniques in amplification. Any one of these techniques can be used for amplifying the sample preparation. The reason for this is the initial would not be sufficient for the ultimate detection of the pathogen. So you need sometimes by using the amplification techniques methods such as PCR to multifold increase of the initial material, so that you would ultimately achieve the detection easily. That's the purpose of amplification, which the reason why you do that. And then you would detect those pathogens which were present in the specimens which are mentioned, by either by real-time fluorescence in real-time PCR, or by gel-based methods, or by melt curve, or by hybridation in case of if it is a DNA microarray, or by electrophoresis if you are doing a simple PCR, or by sequencing the fragment which you had amplified, or sequencing the entire organism which you had randomly amplified and aligned, and then you would identify that what is the pathogen in it actually. So this is the molecular method workflow. So just to summarize this slide, it is always starts with sample preparation, then it goes to amplification, wherein after which you had amplified this initial sample at a certain level, then you would put them for the detection by any of these methods which I had mentioned. Okay, so as I mentioned that there are various molecular methods are available for pathogen detection, pathogen diagnosis and detection actually. So as I mentioned that one of few of the quite interesting ones for our discussion, and also been very widely used by the molecular biology fraternity is them, as I mentioned, are tabulated here. So I would be discussing a couple of these techniques, especially the PCR family-wide PCR method, and next generation sequencing in a quite bit detailed. So, and also show you that how these two techniques actually helped coming up with the complete sequence of SARS-CoV, and then also the same can be applied for the other pandemic situations as well. So family-wide polymerase chain reaction. So what does the family-wide polymerase chain reaction means? As we know, all simple PCR reaction, polymerase chain reaction is simple, that you just have a specific PCR primers, which you would be putting in a sequencing, which you would be putting in the reaction, and along with the target DNA, you would be amplifying it, and you would be detecting it either by gel based, or if it is a real time based, if it is by fluorescence based detection. But how the family wide polymerase chain reaction is slightly varied from the conventional PCR is, you would be designing the primers which would be specific for a family of viruses, okay, and then you would be amplifying them actually. Even if there is any virus which belongs to a particular family, can also be amplified by using these primers, because generally these primers which we use in conventional PCR are very specific and organism specific actually, and also in some cases it is genotype specific basically. So but then when you are using a family-wide polymerase chain reaction, in family-wide polymerase chain reaction, the primers which are more degenerative and which would allow you to amplify any of the viruses which belongs to the particular family, give you a possibility that you would be capturing any viruses, any novel viruses which are coming up due to certain mutations, or escaping from one species to another species as a transmission, etc., would be captured by that. It is an invaluable tool for identification of pathogens, definitely the reason is that, as I mentioned, it would able to cover the entire family of viruses in a particular viral family if you are targeting. And in that viral family, of course, it would be able to detect all the known and unknown pathogens, and it is very rapid. The reason is that as you are using only one primer set every time, so you would be able to capture even tomorrow if there is another virus existing or coming up as a new viruses in that family can also be quickly captured actually. And it also replaces high cost of surveillance studies. The reason is that otherwise you would be spending a lot of money in doing surveillance studies and designing a very high cost methods which would capture those kind of new emerging viruses actually. So once we amplify the PCR product, we put them the PCR product either by sizing or sequencing. We will detect that, and then once we have the detection uh, either by its size, fragment size, or sequencing, then we build the binary matrix and distance matrix of that, and go with the tree construction, that the phylogenetic tree constructions, and would be placing the different viruses or the new pathogen the location actually, okay, by using the bioinformatic approaches. The another valuable tool which is available for the research of novel pathogen discovery in next generation sequencing. Next generation sequencing is definitely an excellent tool which is available and helped a lot in discovery and sequencing the novel pathogens such as the pathogen causing COVID-19 as well actually. How did this help is in combination with the family-wide polymerase chain reaction. People could able to amplify by using the common influenza or SARS PCR primers, which are family-based, family-wide primers, and put them after the amplification in next generation sequencing. They found that the sequence of that is different from the SARS, and then ultimately sequenced the entire region and the entire organism, and then found that the actual organism of the cause actually of the COVID-19. So that's how it happened. I just take you the brief workflow of next generation sequencing. So the next generation sequencing workflow starts from the preparation of clinical specimens. You would homogenize them, and then after homogenization, you can either subject them for the density gradient centrifugation or nucleus digestion and low centrifugation you can use. So if you are subjecting them for the density gradient centrifugation, you would be getting a pure viral particles, which you could further use it along with your your next generation sequencing for the other characterization purposes as also. But then if you are using the nucleases and low centrifugation, then you would be getting a semi-pure or enriched viruses, which you need to, in either ways, either you have a pure virus or a semi-pure enriched virus, as you could do the nucleic acid extraction from them, and then you could put them for the amplification, and then by using the random library preparation primers, you would generate the entire organism sequences, and then sequence and by putting them in the next generation sequencer, you could able to sequence them. Once the sequencing had been done, then you do the data collection, and then you store the data, and then you also process the data for the analysis purpose. So while in analysis, what you do is we try to first make the alignment of all the context which you received, that is the sequences which you obtained on your sequencer. And then you do it by de novo assembly. You will assemble them and try to get the entire structure of the organism and the full length of the sequence of the organism, and then which eventually will tell you that which virus or pathogen it is actually. So once we have the information about any new pathogen, what is the use of that actually? The use of that is in these kind of pandemic situations. Yeah, the use of that kind of information is invaluable. That is that it has to be quickly utilized to develop diagnostic assays so that people can be screened at mass and know that and can be utilized for the surveillance studies, and also know that who are the people that are infected, and to stop the spread of further infection, and also to contain the disease. It is very important that this information is quickly generated and that information is utilized to develop the diagnostic assay. So further in my talk, what I would do is I would let you I will let you know that what are the different stages that we follow at an industry to develop the diagnostic assay by using the information which is available from any new pathogen or any existing pathogen. Okay, so definitely this starts with a design, which means in the design you would be looking at all the available sequences which are at public domains such as NCBI, etc., and you would align them to design your primers which are specifically identify the pathogen which is of your interest. Okay, and then you do the feasibility study quickly, that is that whether your particular pathogen, particular primer which you have designed is suitably picking all the genotypes of your interest or subgenotypes of your interest or not, or whether it is binding to any non-specific organism, etc. You would check actually, and then you would optimize the formulation which you had initially used it feasibility, so that you get a better optimal performance of your formulation to efficiently detect the organism. And then you do your assays for the verification and validation studies. In verification, quickly I could say that you would check that whether the performances of your requirement is coming up or not, and then you will further validate the assay in clinical samples and in clinical trials, that whether this particular assay which you had developed is giving you the satisfactory performance compared to the existing methods or the existing comparable methods actually. And then once the development is completed, you would be transferring it to the manufacturing, and then the manufacturing would take it further forward and make the required batches, and that would be put forward for the commercialization actually. And during this process, the regulatory part is interlinked, and each and every step is closely observed and documented. Each and every step is should be traceable. I would talk much on them in further slides. So one of the key steps in the development of a diagnostic assay is molecular diagnostic assay, especially qPCR based molecular diagnostic assays, is design. Your design has to be very perfect, so that your initial requirement or or achievements which you would like to do can be achieved easily. So that's what. So what you have to do when a design, when you are looking to design a qPCR based molecular diagnostic kit is that the first step what you do is you would look for the literature, and you also, if there is any existence of the literature about a particular pathogen, or if it is not available, then if it is a new pathogen, then whatever the available sequences at NCBI, you try to align them and you get a consensus of that, and you choose the areas which are conserved in a particular conserved and specific to a particular pathogen. Then you design your primers specifically for that region which you have chosen, that which is specific for that particular pathogen, and then you would select those primers and you will analyze those primers and probe sets. So you would do the analysis of the primers and probe sets such as their melting temperatures, their hairpin loop formations, their any secondary structure formations, their GC content, etc. There are tools available which I would not be discussing here, but there are tools available, commercial tools available, which by using which you could able to do this. And once you select the right set of the primers which is fulfilling these criteria, you could proceed with your laboratory part, that is the feasibility studies. So at feasibility studies, what you would do is you would check the specificity of the primers and probes which you have selected. Especially you would check that whether this particular the primers and probes are giving you specific amplification of the entire genotypes which present in a particular organism or not. That is what you would look at in for specificity, and you also check for non-specificity. In non-specificity, what do you do is like you would check the non-specificity, whether the primers and probes which you have designed is binding or amplifying any of the organisms which may coexist along with the particular pathogen in a infection, in a kind of mixed infections or not, or in a closely related genotypes or closely related organisms. That is what you would check. Say for example, in case of acid, if you are developing for HIV, then you would check for TB, because mostly TB is I mean HIV is co-infected with TB as well actually. So that's the reason. That's what you do it, and you do a quick cross check for non-specificity of your particular primers and probes which you have selected. Then you would also check the format which you would wanted to have, whether you wanted to have a very simple format of PCR, or you wanted to have a detailed 2-3 steps PCR. This all depends and comes at the stage of feasibility where you would check. Then in further development, what you do is you do the optimization, you do the verification and validations. In optimization, primarily what happens is that you would optimize the formulation, whether it could be the formulation related to the PCR mix, or the mix which you would be using it for the amplification, etc. Mostly you would be optimizing it to bring it to such level that you would achieve the maximum performance of such formulations. So at product verification stage, you would check for the sensitivity, specificity again, and then linearity, precision, etc. So what I mean, before going to the details of sensitivity, specificity, etc., I would say that at verification stage, you would you would check that whether the design which you have developed is giving you the performance characteristics which require or not actually. So that's what eventually and essentially you will check it actually. So sensitivity and specificity, linearity and precision are very key parameters when it comes to a diagnostic assay. So these are the hallmarks of a diagnostic assay. The sensitivity and specificity has to be very close to 100%. And linearity, when you are talking about a linearity, it has to cover a broader linear range so that you get the quantification of the organism precisely. And also your assay have good precision, that you don't give one time one result, another time another result when it comes to the quantification is concerned. So what happens at checking those parameters such as sensitivity? Sensitivity is something which you refer as the ability of the test to correctly identify those patients with the disease. That means if a patient have a disease, if your assay have a 100% sensitivity, that means it detects the patient, it don't omit any patient who had disease actually. So which means what essentially that if you have an assay of 100% sensitivity, which means you are capturing, you are reporting every patient whom you encounter with an infection is a positive case. So that's what it means actually. And you have to and will be targeted when in the assay development, it will be targeted always that to achieve the 100% sensitivity. That's what it is, the target. And the specificity, what that the specificity tells you? Specificity is the ability to test correctly and identify those patients who don't have disease. Means like it has to tell that if a particular patient don't have a disease, it should clearly tell, the assay should tell that the person don't have it should not give a false positive result. That means if a patient with no infection is coming to you, under subjecting for the assay, it has to come that as a negative result. It had to not come as a positive result, which means it is that if the assay is 100% specific, then it will only detect those organisms which is for which it is designed actually. And then in linearity, as I mentioned, that the broader the linear range, usually at industry we target 5 log above linearity, which means that right from 10 power 1 to 10 power 5 or 10 power 3 to 10 power 8, you get a good precise quantification. That is what at industry we target actually. And 5 log above is the requirement for any diagnostic assays to be developed. And then precision is something that as I mentioned, if you have a good linearity of an assay, then you would also be targeting a good precision. That today if I measure a particular if the assay had been measured for a particular sample and given a quantification, and then tomorrow, or if it is been repeated, then it should also give the closest value possible, which had happened to be the value of the previous finding. So that's what precision looks for. And then assay, as an assay at verification stage development, you would target those parameters fulfilled according to the regulatory requirement, which is different for each of the individual assays and organisms, because in some of the cases there is a requirement of 100% sensitivity and specificity. In few cases, some kind of liberation is there for that requirements actually. So once you verify the performances of the assays, of course, during the development, you would also be validating your in vitro diagnostic assay on few of the clinical samples as well as, but that's more on a pilot basis. you would not be doing it on a large scale, but once the verification stage where you achieved the performance characteristics of your requirement, then you would subject it to the validation, clinical validation studies or clinical trial studies, which we call it in industry, and in which we would compare with any existing gold standard method, if it is a culture based method, or if it is any other available molecular method already in market. We would compare them with that to establish the clinical performance of the newly developed assay, so that the assay can be in reference with the reference of the reference method actually. So that's what it happened at clinical validation studies, and that is also a prerequisite and an important piece of requirement for the regulatory, if it is an US FDA or Indian IVD, which I would be discussing in later in my slides actually. And then parallelly, stability studies are very important when it comes to the in vitro diagnostic assays, because these stability studies play an important role in the performance of the in vitro diagnostic assays. Most of these assays are ultra temperature stored or above minus 20 degrees stored. So we don't even regulators don't recommend any studies which are in short term or kind of like that. They look forward for the real-time stability study data, and the real-time stability study data is the data which is required to be submitted to the regulators. Unlike for few IVD products which are 4 degrees stored, there could be accelerated stability studies, and that accelerated stability studies data is valid, but for these it is encouraged to have a real-time stability study data, and that data need to be submitted to the regulator. So to summarize the entire development before going to the manufacturing, it would start with your design. And you would start with the alignment of the sequences and choosing the primers, and you would once your design is done at bioinformatically on a computer, then you would come to the lab stage where you do the feasibility quickly, that where your selected primer set and probe sets are performing up to the expectations at feasibility, and then you would quickly optimize the formulation for a in vitro diagnostic assay, and then you would do the verification of all the parameters of your interest and the regulatory requirements, and then you validate that the developed products is of intended use, such as if the intended use is to detect COVID-19 pathogen, then that means your developed assay has to do that. So whether it is doing or not, you would do it validation at validation stage by checking with the clinical trial study, and then you would also establish the performance comparison with the existing gold standard methods, and that's where your development ends actually when it comes to an industry. Then once the development part is done, you would be transferring the technology to the manufacturing. Of course, it's very limited time which I have and we have here to discuss in detail of what actually more happens at an industry level. I'm trying to give you a brief overview of what happens, so that one would be very informative that this is what happens at an industry level, unlike in research level, where many of these things, especially in the development part, goes parallel and zigzag actually. Like one can happen, one can do optimization before, and some of these steps comes before and some of them comes after, but at industry level they goes very streamlined and with the existing, I mean, very well laid processes. so that the development goes as expected and as desired actually, and as per the regulator requirement. So when it comes to the manufacturing, the entire development technology, including the technical dossiers and the manufacturing dossiers, would be transferred to the team of manufacturing, where they would be taking it forward, manufacturing the batches of requirement or mentioned sizes, and producing them on a scale which is required for the market. So once the manufacturing batches are there, it goes then further to commercialization. That's where that's how the products get released to the market and for the customers. Actually, that's what. And then that does not help end there. That the development also comes with again constant touch with the market feedback, which could happen. Which could happen like if some of the assay had been developed and not been working with certain kind of specimens, then if the requirement is there from the customer end, the feedback would be taken and then further improved on those aspects as well, actually. And then so the continuous improvement, or you could say continual improvement of the product would be done at a particular periods in industry when it comes to the molecular diagnostic assay. Especially in case of viruses, you would after some time the entire whatever the virus today we would be completely changed into a different virus. Especially happens to HIV kind of infections or viruses where more number of mutations happens at each and every cycle of their reproducibility. Then what happens? Then that time you have to periodically review your design, whether your design is again able to your assay is again capturing all the genotypes and the subgenotypes are not, and then ensure that the assay is up to updated and up to the market for the current strains which are in the prevalent in the particular population and region. That's how it goes. So when it comes to the regulatory requirements, because it's a in vitro diagnostic tool, in vitro diagnostic assays, the regulations are a little different for the in vivo products actually. And then there are at least 3 very discriminate regulatory bodies which are there existing, which could give you the licensing of your molecular diagnostic assays, or assays can be for marketing. One of that is US FDA. Of course, it is in US and is the highest regulatory approval which you would achieve if it is a molecular diagnostic assay, if it required to be. And then we have CE marking happens at European country product, and you could also apply if your product wanted to be marketed in Europe and other parts of the world. And we have Indian IVD, mostly governed by SEDESCO, and then which would help us to get the approval for the IVD. In vitro diagnostic use for the molecular assay which we developed. Currently, till 2016, wherein MDR medical device regulations had not been revised, till that time, even molecular diagnostics were considered exclusively is one of the drug, and it was so cumbersome during that time. But now, after the change of MDR rules, now it's clearly defined. This falls under the category of in vitro diagnostics, and better understanding after the COVID situation. There is a better understanding in terms of what is a molecular diagnostic assay, because of the reason that COVID had given a lot of chance people to understand what is the RT-PCR and what is the molecular diagnostics is at a scale which are not imagined before actually. So a lot of changes had happened at the regulatory front, especially US FDA had also offered emergency use for many of the molecular diagnostic assays which had been designed for the COVID-19 detection. And at Indian regulations, we had our Indian government had come, especially government officials of SEDESCO, had come forward to a quick authorization of marketing molecular diagnostic assays, especially for COVID-19, etc. So to summarize my talk, I would like to tell that it is choosing molecular method which are specifically help you in quick discovery of pathogen is tricky. It's not a straightforward method, and we don't have a general rule that okay, you could pick any of these tools and you need to pick the second tool this and then the third one this. So there is nothing called like that, a general rule or a general strategy, but one had to be very familiar with the tools which are available currently and in future, so that you could be able to utilize those tools and put it in your research, and also you would be able to use them correctly by knowing the limitations and advantages of the molecular methods, so that you would be able to utilize them to reduce the research cost, or by reducing the research time to bring the information available for the industry people, so that people could able to take up for the molecular assay developments, or further industry people could utilize those informations for vaccine development, or people could utilize those information for the drug discovery. So one who is dealing with the molecular methods has to be very clear with the end to end, that the limitations and the advantages of a particular technique, so that they can effectively use those techniques productively to discover the novel pathogen. That's what I could say. So I would say that selection is the key. Method selection is the key in success of the discovery of novel pathogen when you are dealing with especially the viruses. At least for the bacterial novel bacterial detection, there is certain laid methods that which you could able to follow, but for novel viral detection, one had to feel that the method selection is key. And I would propose that especially the family-wide PCR, which I had discussed a bit detailed, and the next generation sequencing methods are invaluable tools to use in that kind of research, whether it could be detection of novel pathogens in pandemic situations, or if you are a scientist or researcher who are interested in dealing with metagenomic studies, etc. So that's what. So I also conclude my talk with the design, development, and commercialization of the molecular diagnostic assays. It's similar to the any in vitro diagnostic assay which you would eventually develop, but then it also goes into phase wise, it goes to business proposal, it comes with feasibility, and then you do the design verification, then you do the validations, and you put them for commercialization. With the feedback, you always will be improving your product.
Thank you.
Yes. So hopefully I had covered all the aspects of what I wanted to share from an industry person, because there are certain limitations on me that, unlike academicians where I could go and talk on endless I mean, more insights. Actually, due to those constraints, I have limited very much myself in going in depth in most of these steps and trying to give a brief overview of those two aspects, which are the tools that you could efficiently utilize for the pathogen discovery, and what happens at industry, especially molecular diagnostic assays, such as the COVID-19 RT-PCR kits, how did they had been developed and they will be developed in those kind of pandemic situations. This is what the objective of my talk, and hopefully I have covered all those aspects.
Thank
Thank you
you
so
for a
much.
nice and excellent talk. So questions?
So is it open for questions? I have few questions from YouTube, so I'll just take
Please.
one. So first question is like NGS platform assays are less in India than compared to real-time PCRs. Like why this is so? Why you have mentioned like that?
I mean, as a technology wise, always again you are talking why means one point is next generation sequencing assays are very costlier. You have to choose them selectively, which is the assay which is suitable for a particular purpose. Let's say, for example, let me try to explain you in the context of COVID-19, where you have to screen millions and millions of people every day. So the technique must be robust, simple, and cost effective. It has to meet all these requirements, unless it would fail as a technique. Like in case of next generation sequencing, it is complex, it is costly, it is not robust as real-time PCR. So what happens is, for many of the infections on a regular basis, and also kind of situations like pandemic, gives you an opportunity to stick to the techniques which are fit into those parameters which I had mentioned, that so that the overall outcome what you wanted to expect is what you want actually. Like in case of COVID-19, it has to reach to mass scale and it has to screen the millions of people. So it had to meet that requirement. That is only possible with real-time PCR, not with the next generation sequencing platform, because the platform itself is a huge instrument in terms of maintenance and in terms of the technical requirements, and also the complexity of technique and the cost made it not a very successful tool to put it in a regular use of or general use of the diagnostics. That's the reason still real-time PCR is a gold standard when it comes to the molecular diagnostics.
Another question, like which organisms you are looking for optimization, verification, and validation? Like you would be interested in going for. So is there any, like how you are picking up among so many pathogens or so many disease molecules, and you have any specific criteria on choosing, or you have what are the organisms basically you are interested in?
Okay, so it all depends on what kind of assay you are developing. Let's say, for example, if you are developing a COVID-19 pathogen detection assay, so which means that at verification validation stages, you should ensure that it would not pick any other SARS family viruses. That's the one thing which you need to ensure your assay won't amplify and give you a positive result with any of the SARS family. And also you should ensure that not only SARS family and other viruses which are flu-like viruses don't be detected by your assay. That is one thing. And another thing what you have to also consider is that your assay specifically detects all the COVID-19 viral strains which are prevalent at a particular point of time or particular region actually, which means that it covers all the genotypes, subgenotypes of latest, so that your assay is up to date. That means that you are not missing any of the genotypes and subgenotypes in your assay. So that's what, so one has to go with the context of the assay, that what the kind of assay you are designing and what are the selected organisms which you need to pick for the non-specificity studies, and which you need to pick for which are the genotypes and
I think
subgenotypes.
people are interested to know what are you have worked with the organisms, what kind of organisms you have worked with.
So I have worked with HBV, HIV, like all. I mean, we worked with all these organisms like TB. I mean, Mycobacterium, and I mean all these viruses and yeah.
Okay, so last question, just I would take that is, what are the methods that are used in evolutionary study of pathogens?
So sorry, can you repeat that question?
So does these methods are used in evolution studies of pathogens as well?
Yeah, methods. So yeah, so methods for the evolutionary studies, yeah, you could use any of those molecular methods like a family-wide polymerase chain reaction and next generation
So I think
sequencing
we are
are best
using
suitable.
multiple targets, multiple set of
Yes.
this thing, gene sequencing. Starting from gene sequencing, we also develop the phylogenetic tree, and we
Yes.
also go for
Yes.
evolutionary studies. So I think all of these techniques can be used together to map the evolutionary.
Yes. Yes, absolutely.
Yeah. So I think there are few questions, but seeing the limitation of the time, probably we'll move on to the next this thing. Yeah. So Professor Sujit Kumar Bhutia, please.
Next talk, the last talk of the day is we have Dr. Bairagi Mallick from Ravenna University. So it's an opportunity to introduce him. So he did his PhD from Department of Chemistry, IIT Bombay in 2007. Postdoctoral research from Department of Biological Science, Seoul National University, South Korea, and his Ramana postdoctoral fellowship from Department of Molecular Biology and Biophysics, University of Iowa, USA. And since 2010, he is working as an assistant professor in Department of Chemistry and Biochemistry, Ravenna University, the old Institute of Odisha, Cuttack. His research interest is on membrane protein-mediated drug resistance and nano-bio interface chemistry. I invite Dr. Mallick for his talk. Let's begin.
Dr. Sujit, thank you very much for the introduction. And first of all, I would like to convey my thanks to the organizing committee members and the convener, Dr. Vismita Naik, and all members for your invitation to share my work, what we are doing in our lab. And since morning, we have been listening many exciting talks. I think it's audible to all of you, please, you don't mind.
Yes, it is quite audible. We can see you. I think from my end, both the things are working.
Yeah. So since morning we have been listening many informative talks, and since when it's late evening now, I am the last speaker of today. So I was wondering where to focus and what.
We are extremely sorry for that. Every time schedule.
No, not exactly. I was looking where to end off and start with my talk, because it's relevant to the because the topic of the conference is drug delivery approaches and molecular medicine, and I have designed my concept of thoughts and work to such a way that so that I will fit to some extent, which I can share something with all of you. So with that, let me share my screen. Okay. Oh. Bismita, can I just 1 minute? I have done one mistake. I have logged in the Safari. It is not taking from the drive, so I need to just log out and work in the Chrome.
Yes,
So then it will
yes,
take.
please, please.
Please don't mind. Yeah. I'm sorry for my small mistake. But
Yes,
now
I think. Yeah. So, are we ready to like? You are comfortable with your settings?
yeah, it seems like.
So you try sharing your screen.
It's, now I have done. Can you see?
Yes, yes, we can see you.
No, my presentation is.
No presentation, no presentation, but we can see you.
No, sorry, what I did. I don't know what happened. I checked it, it was happening. I don't know. See how it's coming. I am.
So Dr. Mallick, in worst scenario, you can send the PPT and probably I can share from my end. That's the last thing.
Yeah. I'm doing that. I don't know what is happening. I'm just sharing with us 1 minute.
So in Gmail, you can just share me and I can download and share.
I'm just sharing with you. Just 1 minute. I don't know what happened.
ବିସ୍ମିତା ମ୍ୟାଡାମ ଆପଣ ଦେଇ ସାରିଲେଣି ଇନଭାଇଟ୍ କରି ସାରିଲେଣି
ହଁ Bairagi sir ଅଛନ୍ତି. It's
I have sent.
okay, so he is not unable to share his screen, his presentation basically.
I don't know why this, because I don't know why it is not, but I have sent you.
Okay, just I'll check and if I can.
Did you receive?
It received. So let's see. Okay. So no, it's not yet shared. It's,
Yeah. Can you please refresh once? It might be, the last time it happened that way. I don't know what happened. I checked.
So have you sent it the email? In the email, did you send me the PPT?
Yeah, I have sent the PPT in the email.
Okay. Just let me refresh it again. Yes, I received the PPT.
Sorry, all of you, because.
Now let me share my screen. So is it visualized by everyone? That's what I would ask. Can you see?
Yes, it is. Yes, it is visible.
Yes.
Yeah, I can see. Thanks, Viswada. I don't know what happened. I was all set.
Okay, so let's go with the presentation. Not a problem. This kind of things happen. Yes.
Yeah, without any delay, let's summarize. I'll go very quickly. And what we want to do is that the topic is that the drug delivery cannot be successful or cannot be fruitful without understanding its drug interaction with the proteins. So that I believe. So I present here from my part of the work lab is mediated conformational dynamics, equally outer membrane protein toll C. It's now Bismillah control is on you. I cannot do anything, so you have to move slides. Please.
Then you can make it full screen first. Then you can keep changing.
So I have summarized my talk to 4 parts. That's introduction, where I'll be talking about the membrane.
So you ask me next.
Antibiotics, and then what is the efflux mechanism and how the consequences of drug resistance, then approaches to understand drug binding to toll C and its effect. Then that will be discussed in that biophysical characterization of antibiotic bindings. Then conformational changes are related to drug efflux. And in the third part, I'll be concentrating on the molecular approach to understand the conformational changes. And lastly, I will summarize my talk, what we have planned and discussed in this topic. Please, next, I have to. So there is some change in the text, might be because of Windows. So anyway, biology of membrane protein is complex, as all of you know, but needs proper understanding to explore the drug binding and drug delivery system. Drug binding and thereafter causing efflux has developed serious consequences, drug resistance, and however, in E. coli, the system that we specifically choose to understand this drug resistance and efflux mechanism. So the tripartite efflux channel, that is ACR, AB, and toll C, is the leading cause of its resistance. So Tolsi being a member of outer membrane efflux proteins, it acts as an exit dot, a protein antibiotics substrate molecules across the E. coli cell membrane. So export of these molecules is evident to be brought through the reversible interaction of substrate specific drug molecules or antibiotics with Tolsi and open for transport. So drug delivery will be effective only if the drug is not resistant, and so understanding the mechanism of efflux can address the challenges of drug development and thereafter. Let's next slide, please. So then what we do is to address our objectives. So we started with the screening of some of the antibiotics. Let me talk. I can talk. So that is issue. I have all these slides. So we started with all antibiotics, screening of the antibiotic through molecular docking approaches, and we choose 6 antibiotic for our interest. And from the docking score, we could see that the G score for the kanamycin is quite high, that is minus 6.5. So we choose that kanamycin was effectively and preferentially interacting and binding with the Tolsi protein. So being a kanamycin is a promising aminoglycoside antibiotic, and that has been extensively, as you know, that has been extensively used in the treatment of E. coli infection worldwide. So it acts on bacteria by inhibiting the protein synthesis, which is essential for its survival. However, the extensive use of kanamycin for E. coli treatment has developed resistance to a greater extent, and the same is considered to be brought through the outer membrane efflux protein. So understanding how the membrane bound or membrane proteins, they are responsible for influx and efflux of the drug molecules, whether it is a drug delivery system or any system which wanted intended to deliver the drugs for its effect in the cell. So it will be a fruitful consequences of understanding the delivery system as well as the drug resistance. Next slide. We choose to purify this protein Tolsi by cloning. Then we did the HPLC to see the purity of the protein. Then we quantify, and then we check its molecular size by the multi-mass spectroscopy. And once we confirm the protein that we have purified, that already been reported, and then we assure ourselves that the protein that we purified is the same protein that with the same molecular mass and with the same activity. And activity we didn't check with any enzyme assay, but however we check through the CD spectra that the secondary structures are intact. So we thought this protein is still inactive to get the bind with the drug of interest. Then thereafter we see the binding study using fluorescence spectroscopy. And as we did the fluorescence spectroscopy with the increasing concentrations of that is kanamycin, we see a concentration dependent that increase of fluorescence intensity with the binding parameters that we obtain from the Stern-Bolmer equation that clearly indicates that the binding is quite specific to the Tolsi protein with a binding constant of 2.9 micromolar and with a single binding site. Next, Ajay, let me keep this aside. So then to extend again to further extended the binding would be the ITC binding study. And in the IT binding study, we could find that the binding of the kanamycin with the Tolsi shows a endothermic ITC profile, the binding association constant of 2.3, and the enthalpy, which is in kilocalorie per mole, is 80, and the corresponding enthalpy as well as the free energy change. So the thermodynamic parameters from ITC binding clearly indicate that the negative drive, enthalpy, and the entropy compensation indicate it is an entropy driven process, and the entropy driven is compensation through the change in delta G as minus, it's a negative value minus 5.96, which clearly indicate that this process is cell driven and with the entropy driven process. Then After this validation of binding through the fluorescence and ITC, we saw what are the conformations, because the consequence of this binding will lead to the conformational changes in the protein motif where the drug binds. So we wanted to monitor what are the conformational changes that occur after the kanamycin binding with the TOL-C. So we perform circular dicrogen study, and both in far and near UV range to see the conformational changes both in secondary structure as well as the tertiary structure. So with increasing concentration of kanamycin from 10 millimolar to 30 millimolar, it's a high concentrations, because in low concentration the resolution was in CD was very less. So that's why preferentially choose the high concentration while standardizing the binding with this toll protein. And we could see that from the table, the fluctuation or the change in the alpha helices and beta sheets in the secondary structure is not significant. But however, the tertiary structure shows some changes, and that might be inhibiting or inducing the interaction with other proteins for its transportation of the drug. But the secondary structure or no significant change in secondary structure could indicate there is a complex that which will form after interaction or binding of the kanamycin with TOL-C is quite stable at these conditions of experimental conditions. Then once we confirm the binding and structural studies, we thought what is happening, because all these are in the solution chemistry. So to get a molecular on atomic level interaction of this two drug as well as its protein, so we thought to do the molecular docking study. And in the molecular docking, we could see that the surface potential of the view of the TOL-C binding pocket actually fit to the kanamycin, and in the and the ribbon diagram of TOL-C show the interaction of dog kanamycin in the active site and the amino acids that are involved in this in the active site for its binding through and making the complex as more stable. And then we check the 2D representation of TOL-C interaction, and there are 5 amino acids which are involved in the active sites through 12 hydrogen network bondings, and they are the different hydrogen bonding length, and the atoms of the residues that involve most in oxygen, nitrogen, and are the, because it's aminoglycoside. If you see the in my beginning, I have shown a picture for the aminoglycoside drug, that kanamycin is highly hydrophilic. So it is naturally convenient that there is most will be hydrogen bonding, and some of the van der Waals forces are strengthening this binding interaction, because the entropy change is less than 0, which indicates there is a preferential and hydrogen bonding, and which is also strengthened to the van der Waals equations. So hydrogen, then after the docking, we confirm that see how this protein is behaving, because it's a membrane protein, and if you take it out, as you know, this membrane protein, we took almost 3 years to purify this protein, and the project was about to finish, and we could able to manage to get some data. The membrane protein was quite flexible, quite when you expose to outside, it always is going to aggregate. So however, somehow we have standardized this purification through POPP lipid, and we saw how the protein is really behaving through POPP lipid bilayers. So this was the dynamics of TOL-C kinase in the complex of POP lipid bilayer, and is quite stable. And with the the beta sheets are quietly embedded in the POPC lipid pair, whereas the alpha helices are quite hanging through down the lipid bilayer. So then, because the consequence of this drug binding has been mediated through the tripartite protein, that is ACR-A and ACR and TOL-CB. So there is always a changes in the conformation within the open and closed domain of the protein, through which it forms a channel for the drug to transport from the one end to the another for the binding to the next protein. So distal end of this protein is, if you see in the A and B, the distal part of this protein is quite embedded with the beta sheets, whereas the that is proximal ends of the protein is quite hanging in the alpha helix. And the C and D are the exterior view of the proximal end of the alpha helix, where you can see one C is in the closed, and mostly in the upper form of this protein remain in the closed domain, whereas the hollow form, always the binding form, is in the open. So this opening and closing of the proximal end of the protein, that leads to the interaction with other binding partners that lead to transportation or the efflux of the drug molecules or antibiotics through this tripartite efflux channel. Then to understand what is the structural that is motion and internal fluctuations that is happening during this binding and its transportation of the drug, we can see that we did a ED there is molecular dynamic simulation, and up to we can see from this, from the RMSD and radiation of gyration spectra of the plot, that the molecule or binding of the kanamycin has a flame fluctuation up to 10 nanometer, but thereafter it is quite stable up to 50 nanometers. So the initial fluctuation could be due to the long range interactions of that is taking place with the molecules and with the reorientation of the domain, and that for which there is a small up to 10 nanoseconds of fluctuation, but thereafter it is quite stable. So it means the binding of kanamycin to the active sites of the TOL-C is quite stable, and the complex that is quite stable at the studied condition that we have observed from this data. Again, we see how the atomic level fluctuations are taking place even when the structural motions of the stabilize through the complex, how the atomic levels are get fluctuated internally. So we can see that the three chains A, B, and C chains all are superimposed in terms of their number of atoms that interacting, and the number of atoms that is getting fluctuation during this domain is oriented or the conformational changes. So almost all these are superimposed with the small minor changes around the 200 to 300 residues. That is some fluctuations in the chain A, B, and C, but the rest of things even in the 0 to 100 residues, that is also, but all these three chains are synchronously fluctuating with each other. So there is no distinct fluctuations of the chains of this protein while binding with the protein. So it means the binding of the kanamycin with the TOL-C protein at the active site forms a quite stable complex, which is again strengthened or supported to the hydrogen bonding calculations. So altogether, what we so far we understood that the binding of kanamycin to the active site of the TOL-C is quite stable through the hydrogen bonding network as well as the van der Waals. Then with the molecular dynamic analysis of the TOL-C, we plot the eigenvalues corresponding to the eigenvector of the kanamycin complex with TOL-C. And the second, the projection of the TOL-C having EV1, that is eigenvector 1 and eigenvector 2 template for the first two principal component, which give an idea that how the initial fluctuations is off, and again it's going to get stabilized around 0. There the fluctuations are quite stable. So the molecule, the complex that is formed, is quite stable. It is not been destructed or deviated before it released from the TOL-C and get bound to the other proteins. So in some literature we have seen there is a large fluctuations. Sometimes it get before it release and get to the next binding partner, it will lost in between, but it is not happening in this case. So we could conclude that the binding of kanamycin to TOL-C is easily facilitating to interacting with the other binding patterns of the channel, and through which the drug is probably efflux out, and that is the consequences of cross resistance in case of E. coli for the specific aminoglycoside drug. So let me, I have gone very quickly, let me summarize what the talk is. This study concludes that kanamycin binds with TOL-C in active sites and brings the conformational changes that we saw both CD fluorescence, CD as well as the ITC data. Then kanamycin binding helps TOL-C to interact with ACRB protein to establish the tripartite ACRB classification channel to expose kanamycin across the E. coli membrane. Our result indicate the preferential binding of kanamycin with TOL-C and support the earlier proposed efflux mechanism through ACRB TOL-C pump, which was established, and the paper was published in late 19th in the Nature. So the binding of kanamycin with TOL-C and the efflux has detected the consequences drug resistance in E. coli strain, and this is additional support to the earlier published results that binding of other aminoglycosides have the similar effect. So we thought kanamycin is the best model for the to strengthen the other reported results. This study needs, again, this study needs further detailed analysis through domain-domain interaction, which we have extending this because the Tulsi is a tetramer. So unless we understand how the domains are playing during this interaction and binding and how they are helping the transport of efflux mechanism, so this work cannot be completed. But still this work, we have this part of work has been recently, we have submitted to Frontier in Molecular Biology under the review. And so, let me acknowledge my financial support from the UGC Government of India and DBT Government of India for the financial support, and Department of Chemistry and Center of Excellence, Ravenshaw University, Cuttack for infrastructure support. And finally, my collaborator, Dr. Tushar K. Beuria, scientist, ILS, Deviation, and Dr. Suman Jha, NIT Rourkela, and Dr. Budheswar Deuri, University of Cambridge, UK. He has done the bioinformatics work, and we have been working very closely for the other 2-3 projects. And Dr. Dilip Basudevan, scientist, ILS, BSR, with whom we are doing the co-crystallization of Tulsi and the cell kanamycin as a complex. And thank you all. I have still, you can see it's 5:30. So even if you give me, I was, so I have finished before you need time.
So let me go back to the original screen because I had shared it.
Yeah, please.
Just hold on for a second. I'm not able to get it.
Timely finished.
Timely. So we are very much happy and thank you that you have.
No, I have no chance to bore all of you because I was get also in stock what happened. Why didn't I could not share, and it's something when happened. I in fact get stressed out. But anyway, it was. I hope I have some information to share that I have done.
So I think Sujit sir, the session is open for a discussion. We have a little bit.
I have not seen in the chat no question. Please help if anybody have a question from YouTube.
So YouTube, actually I was sharing, so probably I have not seen the YouTube channel to see actually the questions. But I think I have some few questions if I am allowed to ask questions.
Yes, please. I'll be delighted to my pleasure to answer you.
So you actually talked about the efflux of drug resistance. So we all know that there are various channel-based mechanisms by which we can target not only for kanamycin, not only for various kinds of drugs, and it's very much applicable in case of cancer also. So any particular any mechanisms where we can actually change some modality on the surface and we can change this throwing out of this resistance, basically throwing out of the drug from the cell itself. So if
no, there is a lot of mechanism that have come to the modifications of the surface bound proteins, particularly where you can, because the most important thing is that the retention time of the drugs. Unless the drug retain for a period of time where it can release and get its effect, then it will be automatically being a foreign invader, it can be taken away, so it will get just that we understand the reflux mechanism. So to retain that drug inside the cell is the most viable point to stop this efflux mechanism, and that only could be happen if we design some molecules which are preference. They are not competitive binding, but they can compete for the same binding, which they will sit, and so that the drug may be interacting with the same person with the alternative, but that cannot get efflux out in the same time without before it retention time. So about cancer biology, we have not tested, we have not concentrated on any of the channels. But we were working in some proteins we were working on some proteins which are relevant to the breast cancer, which is undergoing. So maybe sometimes we'll have those data to share. But as of now, we have only concentrating on the bacterial system. So not much of data or information about the cancer biology, flux mechanisms.
I think Mona Lisa, madam, she is with a question.
Yeah. So hi, this is a very nice talk, and I have few questions. Is there regarding the channel only? So is there anything known that this kanamycin it can interact with any of the channel protein in the human being, in any of the organ, any sensory organ?
Yes, ma'am. We checked that a paper in Cornasic in Nature 2009, which is quoted from there also. They have shown that they have like CLC channel and other channels. They have interaction with, through that. I can share that paper. The CLC channel, the chloride channel, through which they have shown that this interaction, this interact and bind to the.
It is a nature paper or those baby natures like nature or pure nature? Nature?
No, it's pure nature. That's a crystal structure.
Pure nature.
Yeah, it's a crystal structure, nature paper. There they have shown the channel what.
But no interaction with sodium channel.
I have not searched for that, but yes, that's true. But it has written some other papers I have seen, but I have not thoroughly seen. But it is mentioned there that it also interact with the particular, ma'am. It's a cations, particularly they are ion channels. They are interacting with the kanamycin. So because it is highly hydrophilic, if you see the structure of kanamycin is highly hydrophilic, and most of this time it is interacting to the hydrogen bonding, and the van der Waals interactions are the preferential interaction. So as the hydrogen bonding the strengthening, so there is possibility that the hydrogen network, because it substantially destroy the hydrogen network around the molecules and it preferentially go to the bind to the hydrophilic patches of the protein. So that's why it is, they have mentioned that as the chloride channel is also ion channel, so it's binding, it must be binding to the ion sodium channels that.
Yeah, one more question. Is
Yes,
there
ma'am.
any such data from other aminoglycoside like gentamicin and neomycin?
Ma'am, we thought categorically we use the string database to see the initial network, how this interacting with this we choose the 1020 aminoglycosides to see the string based interaction which is really interacting. Then we found a few, then we choose for the molecular docking how the score is. So we have specifically not in the broad sense, we have not taken a large number of amino acids, but the aminoglycoside. But initially we choose 10, but to put in the and search in the string database, and from there we conclude the 6 has some interaction directly or indirectly. Then we did the molecular docking screening from the initially how they are, what is the docking score for that, and from that we found that kanamycin has a highest docking score of G value, so that's why we choose this might be specifically interacting with in the specific this system.
Thank you.
Yeah, it's my pleasure, ma'am.
So, if I can take on one more question, I will just would like to know what is the role of this protein folding, like partially folding and totally folding, and all these scenario of proteins in disease development and how we can modulate it.
So you
basically I'm linking.
okay. Then I don't want to. Okay, protein.
So you talked about some protein foldings are happening in between, and this protein foldings. Maybe I would like to know what are the things we can take control, because in body it keeps forming disordered proteins.
You know that active forms are the folded forms, but the question comes. What is the strength of that active form? Because some proteins even remain in the active form, they behave differently. So unless we have a sequential data where what is the recently this the previous Mr. Verma was explaining, if you use the active form of only, then we can form some formulation or design some pathway to get the targeted disease. But here in the case, unless if there is a degenerated proteins, they are absolutely gone, because again Professor Amulya Panda also explaining how you degenerated protein make it. Many people are now, suppose I get a degenerated protein, we are discouraging or we are get afraid of how to get. But if you can refold them through a new developed process or modified process and make them active, then many problems will be solved. But the thing is that in case of membrane protein, it is so sensitive for us. We tried nanodisk, and it was quite expensive. We put the protein in the that nanodisk membrane, and we tried to purify, but we could not. So sometimes the disordered proteins, to get them in active form and through different means of solvents or the systems, sometimes it's difficult. But however, there are some systems which can make them more active. In the active form, we can address some of the disease, whether how they are behaving towards specific. Maybe I'm not that specific to answer all your question.
So I'll just say, with your understanding and knowledge, like, is that disordered proteins which are partially folded or disordered proteins which may further lead to manifestation of a disease. So is there any scope to isolate them and screen them before they really manifest, so that we'll be aware and we will be taking precautionary measures?
Yeah, of course.
Is there any screening methods are there?
There are some proteins which are not even in the unfolded state; they get purified, and so we can them again, or the inclusion bodies, they go again, that we have to purify as unfolded using urea or any denatured conditions. Then again refold them to the active forms. So there are those proteins which go to inclusion body, they may not be in the active form, but they usually go in the state where they can be recovered through a chemical process. And once you get recovered, then that can be made to active forms through the folding of mechanism. So once you get folded protein, then you have to check its activity through activity assay, or again with the confirmation what is the structural constituents of the secondary structure, it's behaving, it's intact or in active form or not. Then once it is confirmed, then we can plan for some work with that protein.
So I think it's a lot of discussions, maybe further discussions.
I was expecting one question from Dr. Suman Jaivi is there.
I don't see him right now, probably. I told him, but he has some other engagements, yes, and it's already going to be 6. Suman sir.
I'm here only.
So
I was just
actually,
listening.
even I was
You guys.
expecting a question from you. I was expecting your interaction, basically.
No, I already know everything what he's doing. That's why I didn't have any question. So, ma'am, to your question that you asked in the end, like whether if they in a cell, if there are some intrinsically disordered protein which manifest the disease, I would just like to some sentence to what Bairagi sir has said. Like till now, we don't have any diagnostic mechanism to detect them at very early stages, where we can say, yeah, it's going to have some disease like Alzheimer, Parkinson, or something like that, right? Because the proteins which cause these diseases, they are intrinsically disordered protein. So in our cell, almost 30% of proteins are intrinsically disordered protein, which have multiple targets to work on. But to diagnose them, whether they will go in disease or will act normally, it's very hard to predict. And so early diagnosis, that's the main reason why early diagnosis of Alzheimer, type 2 diabetes, Parkinson's, mostly protein disorder diseases, misfolding diseases, are very hard to diagnose at very early stages. But at late stage, when symptoms appears, when you see the neurodegeneration or decrease in beta cell, pancreatic beta cells, then by looking at the symptoms, you can diagnose. So people are working in this area to diagnose them at very early stages. So it's in very naive stage to
But
to
in
have the
that
diagnostic
direction,
tool
actually,
for
I
this
think
kind of thing.
the research should go on. So for early diagnostic methods, I think if possible, if we can target these proteins partially disordered.
Actually, that's a good approach to understand the early diagnosis of disordered proteins. But only thing is that how to detect them and make them, at the initial stage, then that will solve many problems. So that is a real constraint in the disorderedness of the protein. Late stage, many
That's
people.
the main problem.
So I think with this we have talked a lot and we have learned a lot from each other. I believe our students, our viewers will definitely get some benefit and understanding from the talk and from the overall session. I thank you everyone being here, and I really thank you, Dr. Bairagi, Mallick, because I know you from a very long time, but it was really a kind of you for accepting my short notice.
You make me engage for last evening to till evening by preparing all these things at the last moment. But anyway, thank you
I
for.
didn't had a chance to see you live talking and giving a presentation. So I am delighted. I am very much thank you for it.
Thank
And
you.
of course, it's very informative. So I know that students might have enjoyed and learned a lot from you. Thank you, Suman sir, for being here, for being adding things to it.
Suman.
Yeah.
Pleasure is all mine, ma'am.
Thank you so much, everyone here, present here. So with this, we'll end the session. And tomorrow by 9:30, the sessions will start, but people can join a little bit early, 5 minutes to 9:30 a.m. So we'll try to be stick with the schedule by starting by 9:30, and we'll try to finish up on time. So with this, I'll just close up the session, and I thank you, everyone. We shall join tomorrow 9:30 a.m. Thank you all. So bye for now, everyone. Bye for now. Okay. Thank you. Thank you so much. Okay.